Okay, right. Good afternoon. Yeah, so after a bit of a break, neither right, had to. I thought of giving you a bit of a holiday last week, and but as with any holidays, we are going to have small cover-up sessions at the end. Okay, so maybe half an hour, we are going to cover up that week as well. Right? So that's for the last week holiday. And I know, right? I know it's going to sort of hinder your plans if I had the class on Thursday, right? Thursday in the there was a holiday, right? So Friday, if you had any class, we are going to cut that. So with the Saturday and Sunday, nice long weekend, right? So that's what happened everywhere. Even in our batch, everyone went home on Thursday, and Friday we had to cover for them as well. Okay, so everywhere that's the situation. Anyway, so last week holiday we'll be covering slowly as small extra sessions going up for about 7:30, right? So if you didn't inform your homes, did you inform? Right. Chuttak ada maybe 7:15, 7:20 indala next week onwards we'll start covering up, right? Because next week results are going to come, right? You know, yeah, this result was like කාලයක් තිස්සේ එන ගමන් තිබුණේ. Online people, can you hear me? Yes. So, as people are joining, definitely this week will come, right? Next week. Next week, okay. Definitely will come, ne? Okay. I have my congratulation post already made. So, I was planning to put it this week, but it's been postponed. So, I have already planned for your best results. Anyway, right? I have talked about this, right? It's a whole different game, A levels, right? It's a whole different game. What you need is just to get through O levels, just to get through. It's a sort of a, what do you say, it's a hurdle, right? Then you have a race. After getting through the hurdle, you have the competition, proper one. That's this one. Okay? Because in O levels, you don't get a island rank. Island rank, they say there's island first, second, third. Nobody knows how it's decided. Ne? Island first, decide. No. It doesn't matter, right? Because in A levels, everyone has a rank. Everyone has a rank except if you got all 3 Fs or 1 F pass, you are not going to get a rank. But everyone who passed the exam is going to get a rank. Ne? There's an island rank number 1. In your stream, there's going to be lower ranks as well. Right? That depends on the EZ score and everything. Okay, so that's the A level situation. Anyway, we'll have a small chit chat as we go along. Anyone who didn't get our 2 tutes, that's we are going to continue these 2 this month as well. Online people, hope all of you have got it. I'll be sending the PDF to the Zoom chat, so please download it if you haven't gotten delivery. Anyone who can't hear me? Any issue? You can hear me, right? Okay. All right. So, okay, we'll get into it. Right? It's already. We have to start. So, where did we stop last week? Right? Where exactly did we stop last week? We are done with the cell. Are we finished? I mean, have we finished talking about what is inside? What is happening inside the cell? Done. Intracellular structures are done. Yeah. Everything to know about inside of the cell, we know, right? Everything that is inner to the plasma membrane, we have covered. Did we go outside the cell? Yeah. Did we go to extracellular structures? Cell wall we discussed. Yes, cell wall we discussed. Then today we have 2 more structures to discuss. That is cell junctions and then the extracellular matrix. Afterwards, then we are going to start as advertised cell division, the process we are going to start. Okay. So that is going to be our next big step, and as I keep on telling, this cell division, it's very important. I have sort of found this in my 2026 batch because we just finished this sort of molecular biology, and genetics was done a long time ago. But when I'm doing those lessons, I found out they are sort of basic cell division knowledge is not there. Simple things, how the chromatin becomes DNA, how the DNA is separated during cell division and goes into the two cells. And in meiotic cell division, how there is mixing up of DNA. මෙයාගේ DNA මෙයාට යනවා, මෙයාගේ DNA මෙයාට යනවා. That sort of basic understanding is not there. So at that time, I don't have the time to explain how basic cell division happens. So that's why I keep on telling. Then just because you are sort of in your sort of the third or fourth month of A levels doesn't mean චුටි චුටි ඒවා විතරයි කියලා, right? You have to think ahead, you have to plan ahead. දැන් ඒ කාලෙට ගිහිල්ලා මේක තේරෙන්නේ නැති වෙන විදිහට කරන්න බෑ, right? We have to teach with a plan, right? So even though you are still at the very beginning, we have to teach in a manner that's going to help you in the future upcoming lessons as well. That's after we have started that section. So we'll quickly get through these other extracellular structures. Okay, so come to the resource. I hope at this point you have figured out the resource. Online people, all of you have got the resource book, right? PDF. Okay. So cell wall we discussed. Did I explain the functions of cell wall? That is in page number 38. Functions of cell wall. Tuts තියෙනවා නේද? Yeah. Functions of cell wall we discussed, right? Did we not? Okay. So if I quickly recap, because the next point, I mean the next section that we are going to discuss, that is cell junctions. ඒකටත් අපිට පොඩ්ඩක් we need a bit of knowledge of how cell walls form, how cell walls are developed, and how they are maintained and everything. Okay. Right. So if you take a plant cell, because in this lesson we are mainly focusing on the cell wall of plants, okay? Because if you take, what are the other options of cell walls that you can have? What are the other options for cell wall? Plants are the cell wall එකක් තියෙන්නේ. No, right? If you take fungi, they have a cell wall made of chitin. If you take bacteria, they have a cell wall made up of peptidoglycan. There's a variety, polysaccharides and proteins. There can be a variety, right? But here in this lesson, we mainly talk about the cell wall of plants, right? So I'll quickly recap, okay, right? Taggala will come to cell junctions, okay? So if this is a typical plant cell, I think I showed you with the software last week as well. Typical plant cell, it has a sort of a polygonal shape. Yeah. What is this cell wall that we are drawing? This is the primary cell wall. So inner to the primary cell wall, what do you have? Inner to the primary cell wall, make a living plant cell. Okay, this is a living plant cell. You are going to have a plasma membrane. Yeah, it's a living cell, right? So the plasma membrane is there. Then inside you have, usually since it's a plant cell, you have a big vacuole. Yeah. Then you can have a nucleus with DNA inside, and you can have some inner membranes, right? Endoplasmic reticulums. You can have mitochondria. Then you can have chloroplasts. The organelles are there. This primary cell wall is very good, right? It is permeable. Things can go through. Things can come out. It's a nice supporting structure. What's going to happen if you are destined to have? You will going to develop a cell wall, sorry, secondary cell wall later. right? ඒ වගේ කේන්දරේ තියෙනවා වගේ මේ cell wall එකේ තියෙනවා in its sort of let's say programming, it's going to develop a secondary cell wall later පස්සේ කාලේ. Okay, what's going to happen? The secondary cell wall is going to develop inner to the primary cell wall and outer to the plasma membrane. මේ black color, this is the secondary cell wall. Online people listen. හරිද? This is the secondary cell wall. We are talking about extracellular structures.
Yeah, කියනවනේ මේ secondary cell wall කියලා.
What is the how do you describe this secondary cell wall? It is impermeable, it is rigid, right? It is, I mean, it can be composed of cellulose, but it contains hard, rigid substances such as what? Lignin, suberin. Suberin is a lipid, no? Right? So they are going to make this a water impermeable, rigid structure. Okay. එතකොට මොකද? Everything that is inside cannot get material from the outside. වතුර එන්නෙත් නැහැ, nutrition එන්නෙත් නැහැ, nothing. Okay. The secondary cell wall is a death sentence. Okay. The secondary cell wall is a death sentence. So actually, any cell with a secondary cell wall is a dead cell. මැරෙනවා. Then it is going to function purely, I mean mainly as a supportive cell, support cell එක. And it makes a nice tube. The make a මැදින් වතුර අර මේවට එහෙම it can go nicely. Xylem sap can go nicely through this empty cell. It's nicely reinforced on the high වෙලා තියෙන because of the secondary cell. Okay. So that's what we talked about last week. Okay. With that idea in mind, okay, we can talk about. Did I read the functions? Did we talk about the functions of cell wall? No? Okay. Come to page number 38. Okay. Come to page number 38. You can see functions of the cell wall. Protection and support. Okay. So these are functions common to either primary or secondary. Okay. Allows development of turgidity when water enters the cell. I remember I taught this. Yes or no? Okay. What do you mean by development of turgidity? මම මෙහෙම example එකක් ගත්තේ නැද්ද? I taught think of it like if your plant cell, think of it like a water balloon that is put inside a cardboard box. I told you that example, right? Yes. මොකද මේ turgidity කියන්නේ, right? What do you mean by this turgidity? Is like something like this. If your plant cell is similar to a water balloon inside the cardboard box. What do you mean by that? This water balloon is the cytosol, right? The living part of the cell put inside a cell wall. So what you can do is when water enters into this cytosol, when water enters, right? This cytosol is going to get bigger and bigger, and that sort of enlargement is going to put a pressure on the cell wall. That's Newton's third law. There's going to be a opposite and equal force backwards, right? So the cell wall is going to push back on the cytosol. Then what's going to happen is that this entire system is going to become pressurized, a rigid system. There's going to be a force on the cell wall by the cytoplasm and the cytoplasm on the cell wall. That is called turgidity, right? That is like the cell wall, sorry, the plant cell is pressurized, stiff. That is how these small plants stay upright. Now, if you take like a big woody tree, right? Take a කොස් ගහක් වගේ ගත්තොත් they have a massive trunk. Inside there's wood. So that is why it's very tall and very strong. Small plants, they are not woody, right? That is how they stay upright by this sort of stiffness that is provided by the cell wall and water. Okay. The turgidity story we are going to talk about in a lot of detail when we are doing the fourth lesson, plant form. Okay. Then what is the next function? Prevents bursting during turgidity. Then when the water is inside, it's going to be pressurized. There's always a risk of this water balloon bursting. Why it is being stopped from bursting? Because there's a supportive structure outside. But if you take one of your cells, animal cells, there's no such a support system. So our cells, animal cells, can become burst. They can burst easily. Right? For example, our red blood cells can burst easily. Right? So that's why we have to maintain our sort of blood. Can't hear. How's that possible? Anyone else having that issue? Yes. You can hear me, right? Okay. All right. Particularly, your blood cells are very susceptible to this bursting. The make right prevents bursting during turgidity. You don't have a cell wall, right? If your blood solute concentration changes, your red blood cells can burst very easily. The red blood cells have a very unique shape like this. No, describe current your red blood cells as a biconcave. Okay, it's a very unique shape. This can burst very easily because you don't have a cell wall to support your cells. Okay, right. Next function, limits and control cell growth. Again, because this cytoplasm is put into this sort of rigid structure, it can't grow excessively, right? There's always a limitation by the cell wall. Then, component of apoplast pathway. Okay, apoplast pathway I'll explain when I'm talking about another structure called plasmodesmata. I'll explain. Maintain cell shape, of course, right? Because it's a rigid structure. Hold the plant up against the force of gravity, that is by the turgidity. Okay, clear. So this was just a reminder of what we learned last week. Ne? Medica we did last week. Okay, right. Today we'll go ahead with the proper topic that we have to tackle today, that is cell junctions. So if you come to your diagram tute, page number 6, we have I have given you the set of diagrams that you need to follow as I teach you this. Cell junctions. Page number 6 of the diagram tube, this one. Online people, I have sent the PDF to the Zoom chat. Quickly download it if you haven't. So that is the big topic for today. Okay, right. Cell junctions. So if you are a multicellular animal, that means you have more than one cell. Your cells are not like tarha with the neighbors right? They are always communicating because they work together to make you, right? They work together to make tissues, tissues make organs, organs make organ systems becomes the organism, ne? Right? So cell to cell communication, cell to cell connections are very, very important, right? That's why we have to pay attention to these cell junctions. They are very important in upcoming topics such as muscle contraction and other small topics a lot. We are going to touch them, right? So let's read about them first. Come to the resource. Cell junctions. Neighboring cells join, interact, and communicate via sites of direct physical contact. Okay. So you should always a cell junction is a place where neighboring cells communicate. Okay. So in your syllabus, you have three main types, and we have another special plant cell junction to talk about as well, right? So you have three main cell junctions to talk about. Cell junctions are structures at which cytoplasms of adjoining cells are joined. Can you highlight those two sentences? Okay. Neighboring cells join, interact, and communicate via sites of direct physical contacts, and the one below, structures at which cytoplasm of adjoining cells are joined. Please highlight that. Okay, we are exactly, can I know exactly where we are? We are exactly on page number 38 of the resource. Okay, exactly at page number 38. Okay. Right. So those two sentences get together to form a definition. Right? Later I'll give you the definition with the combination of those two sentences. For now, let's read. They connect the internal chemical environment of adjacent cells. There are three types of cell junctions in animal cells. Either we have to talk about desmosome. Sorry, first we have tight junctions, then we have desmosomes or anchor junctions, and then we have gap junctions or communicating junctions. Three types of cell junctions we have to talk about, right? So let's read about the first one. Tight junctions connect the plasma membranes of adjacent cells tightly bound by specific proteins forming continuous seals around the cells. That's a definition. what is a tight junction කියලා හැවුවොත් ඔන්නෝක තමයි ලියන්න. Got it? They connect plasma membranes. That's a very important word that you have to write. So tight junctions. Tight junctions. They are like a silicon done, right? When you if you have a new bathroom, right? You waterproof it after you fixed a new sink or whatever, or a new window mocker. There's a silicon glue, right? That you use to make the structure waterproof. So in your body, whenever you need some waterproofing, we have a tight junction. So if you have two adjacent cells like this, then there are two cells that are adjacent, right? So imagine this is your skin, okay, and you are in a swimming pool. Then swim. So the skin is not like this; it's more sort of multi-layered. Okay, just for example, okay, you are sitting on a swimming pool, right? So there's water outside, and inner to that you are going to have other connective tissues that is underneath your skin. Then this water is going to come through. There are small gaps between skin cells, right? This water is going to come through. Your skin is a watertight barrier, no? It's a watertight. Pool leke behala iti anga athule wathura pirenne, right? It doesn't happen. You don't get water inside your body, right? That is because your skin epithelial cells are connected by this waterproof junction, sorry, watertight junction that is called tight junction. So they are formed by specific proteins that are going to connect the plasma membranes together. The plasma membranes are connected tightly so that this outside liquid can't get into the inside. Okay. So in your diagram tute, I have given you exactly that, right? Can you check the first diagram, page number 6? Can you see in a tight junction, there are two cells on the two sides. Right? It's like a stitch connecting the two plasma membranes. You have two cells on the two sides, right? Can you see the plasma membranes are tightly connected? Okay. So whenever you are learning one of these junctions, what you need to know is how do you introduce them? So how do you introduce it? They connect the plasma membranes tightly, forming continuous seals. That's the nice word that you have to use. So it's a continuous seal. Okay, so it's a continuous seal. And then what are the examples that they have? Prevent leakage. Continuous seal that is preventing leakage. Prevents leakage. Okay, prevents leakage. What is the example that you need to remember? Skin epithelium. Right? You can find this type of tight junction in your digestive system as well. In your digestive tube system, such as your small intestine and large intestine, as food travels through this tube, right? Whatever you are absorbing, for example, if you had some chicken, the amino acids after being digested should go through the cells. It can't, so the pieces of chicken can't get absorbed like through the gaps in the cells. They have to come through the cells. Okay, so that's why you have this sort of tight junctions in your digestive system as well. Okay, so for now, remember skin epithelium. We'll later add digestive system as well. Okay, we'll later add that because in questions they have asked this. Clear about the tight junction? Nothing much. Watertight, continuous, seal. Okay. Next, we have anchor junctions or desmosomes. Anchor junctions or desmosomes. Given about mechanically attach the cytoskeletons of adjoining cells by intermediate filaments for strong binding. That's a definition. Highlight that. Mechanically attach the cytoskeleton. So there are keywords there are some keywords. After highlighting, underline cytoskeletons by intermediate filaments. Okay. So next we have anchor junctions. Anchor junctions, we call them by a sort of a serious word, desmosomes. The best way to understand how they work is by imagining where they are found. What is the example that they are given? Muscle tissue. Muscle tissue. So your individual muscle cells are generating this force, right? Then gym eකට ගිහිල්ලා bicep curl කරනවා කියලා හිතුවා. That dumbbell is lifted by your individual muscle cells, okay, right? So the force is generated by the muscle cells, but there's a chance that the muscle cells can be pulled apart, right? Like a දිගට muscle cells නේ. So for example, if you take your bicep, it's this much long. If you take your quadriceps, it's this long, right? It's not composed of one long cell. Cells are connected one after the other. The muscle cells, අපි ඒවට කියන අතර muscle fibers කියලා, right? We actually call these as muscle fibers. They are actually cylindrical cells. මම මේක පෙන්නන්න, right? They are actually cylindrical cells that are kind of elongated, right? Muscle fibers. So the force of muscle contraction is generated inside the cell. But when they are generating that force, there's a chance that they will be separated. No? And they can be pulled apart. How is that prevented? By the tight junction. So, inside our cells, we talked about this last week, we have an inner sort of a supportive structure, particularly for your animal cells, that we call as the cytoskeleton. We call as the cytoskeleton. Cytoskeleton, microtubules, right? Actin filaments, there's an inner skeletal system, right? At the anchor junction, this cytoskeleton of one cell is connected with the cytoskeleton of the other cell by intermediate filaments. Tightly connected. Okay. So when these muscle cells are generating force, they are not pulled apart. Okay. Very strong connection. Okay. So that's why you can find it in muscle tissue. So method the keywords are they attach cytoskeletons by intermediate filaments. Okay. So what's the difference between this and this? මෙයාගේ priority එක is to prevent leakage. මෙයාගේ priority එක is strength. Okay. This priority is strength. Because you can see by intermediate filaments for strong binding. Okay. Right. Finally, we have the last type of junction, that is gap or communicating. Gap junctions or communicating junctions are kind of like between your house and the other, sort of your neighbor's house, you have sort of a opening. So you can, right, have a sort of a exchange and communication, right? That sort of a communication you can maintain, as the name says. Okay, right? Yeah, before that, can you see this is the diagram of a desmosome? The second diagram, online people, can you see? Second diagram, there is a desmosome. अभी मैं इस तरह आपको मैं लिए ना, okay? For now, visualize it. So can you see on the two types, on the sort of the two sides, you have two cells, and their cytoskeletons are coming together, and at the actual anchor junction, it is like tightly connected by intermediate filaments. Okay. Then I have given you a D2 that is like a desmosome, right? D for desmosome. Can you see how it can be seen in an electron microscope? Maybe gray color, yeah, electron microscope. What kind of electron microscope do you think this is? It's a transmission. No? Yeah, you can see only a cross section, right? Cross section and transmission. Got it? Why is it black and white only? A part of it. That's how electron microscopes are. You can't see colors. Okay, you can put artificial colors, but later, right? That's not routinely done. So that is desmosomes. Okay, come to gap junctions or communicating junctions. Read. Provide cytoplasmic channels from one cell to the adjacent cell. Gap junctions consist of special membrane proteins that surround the pore through which ions, sugars, amino acids may pass. They allow signal and material exchange between adjacent cells through direct connections. Harry. Highlight. These are මෙතන highlight කරන cytoplasmic channels from one cell to the adjacent cells. That's a definition, right? Part of the definition, okay? After we'll write down a proper one in the tute, okay? So they work like this, right? So there's one cell like this. There's a cell like this, and this is the other cell. Your neighbor's house and your house, right? So if you are going to have a communicating or a gap junction, it is not just a hole. I'm just going to draw one communicating junction, right? There's like a group of junctions at the same place. It's not just a hole, right? Right? There's a regulatory mechanism. That is because it is composed of a special group of proteins, right? It's actually a ring of protein. It's actually a ring of protein like this. If you magnify it, right, it is actually composed of you have proteins, and those proteins are sort of extending so that you have another opening on the other side, right? It's kind of like this. These proteins are able to regulate whatever substances that they want to exchange. Right? So that's the sort of the distinction I want you to have. Right? So again, what are the things that they have given in your resource? Ions, sugars, and amino acids. Okay. So these are the ways that we describe. Okay. They are cytoplasmic channels. Okay. They are cytoplasmic channels. Okay. These are the keywords that you need to know. Then they have special membrane proteins, okay, composed of special membrane proteins. And what are the things that are allowed to go through? What are the things that have the permission to go through? Ions, sugars, amino acids, right? This differs from one cell to the other, okay? The place and everything. They allow signal and material exchange through direct connections, okay? Ions, sugars, and amino acids can pass. What is the example? Heart muscle and animal embryo. Heart muscle and animal embryo. What do you need this sort of a communication in your heart? Why do you need that? Because your heart is not just a pump, right? How, I mean, I want you to visualize your heart not like a sort of a place where muscle contraction pumps out blood. Your heart is a pumping bag. Right? It's a pumping bag. You fill blood into that bag, and that bag squeezes. Then blood is sprayed out. Okay, that's how the heart works. So blood fills into the heart chambers, then the entire heart contracts. Right? It sort of twists itself, or twist. Then all the blood that is filled inside is like squeezed out, right? So for that to happen, all the heart muscles should work simultaneously. All the heart muscles should work together, right? So I think in O levels you learned that all the heart muscles are interconnected by intercalated discs. Have you heard? Heart muscles are interconnected. That is also to make the heart contraction a nicely coordinated one. Contracted. Okay. If your heart muscles are not contracting all together, that's a disease, right? You can call them as arrhythmias, abnormal heart rhythms. Okay. Right. That's why you need to have gap or communicating junctions in cardiac muscle. Okay, so you have in heart muscle, I think it's called cardiac muscle, and animal embryo. Embryo means when you were a small ball of cells, mataka the morula, blastocyst, right? There were some developing stages, mataka. First, you were just a single cell called zygote. Okay, then you became a ball of cells, mataka. As you are floating towards the mother's uterus, then I don't think you had up to. Did you have blastocyst? The word blastocyst, tibba, 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 tibba. There's a cavity another sort of. It's still a ball of cells. You can't see any hands, heart, face, or limbs, legs, or bowl of cells, right? So in that bowl of cell stage, all the cells should have a communication because they haven't decided who's going to become what. How the brain again, how the heart again, who's going to become lungs, who's going to become the digestive system, no idea, right? All the cells have the potential. So in that embryonic stage also, you have a very rich supply. I mean, the embryo has is filled with communicating junctions. So embryo, okay, got it. Okay. So come to the diagram tute, page number 6. Okay. So for the first diagram, it's already labeled, but it would in locally and a tight junction killer, right? It would in locally and tight junctions. Okay, tight junctions. Then on the side, we'll write down some points. Okay. On the side, write down form continuous seals. Okay. What points take clear, right? On the side, form continuous seals. Right. On the side, there are some space, right? Form continuous seals. Second point, prevents leakage. Okay, write down. Prevents leakage. Prevents leakage of extracellular fluid. Of extracellular fluid through intercellular space. Prevents leakage of extracellular fluid through intercellular space. Okay. Through intercellular space. Okay. So if you are sitting in a swimming pool, that's how water doesn't come into your body. That's how in your digestive system, as all the things travel through your small intestine, stomach or large intestine, it doesn't get absorbed, right? Food goes through the tube only. Only the nutrients are absorbed because they travel through the plasma membrane, not through these gaps. Gaps are lingera chicken calibadine, right? Okay, that happens when you have a disease, right? If you have a sort of a ulcer or a wound in your stomach, right, due to recurrent gastritis, we are, I mean, they when in that situation we say you have a perforated bowel, hilakti. In that situation, everything you eat can go through that and cause a massive infection in your abdomen. Right? Peritonitis, very serious. Okay, so that's how your tight junctions help you survive. Got it? Then the second diagram, okay, second diagram I don't think, okay, itana desmosome kila dila thiyena, right? Label the second diagram as desmosomes. slash anchor junctions. Okay, desmosome slash anchor junctions. Then on the side, write down mechanically attach cytoskeletons. Mechanically attached cytoskeletons by intermediate filaments. Either mechanically attached cytoskeletons by intermediate filaments. Okay, for tight junctions, examples you examples දෙක ලියන්න. Okay, skin epithelium විතරක් ලියුවා නම් ඇති දැනට. Okay, write down the example for tight junction and also for anchor junction. Okay, muscle tissue is the example for anchor junction or desmosomes. Then third one, label the third diagram. පල්ලෙහාම තියෙන එක. Okay, the lowest diagram. There's no label there. Okay, so label it as gap junctions or communicating junctions. Label it as gap junctions or communicating junctions. You can see there's also a electron micrograph given there. Can you see the place where you have a nice sort of series of communicating junctions? Can you see? There's like dot 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 dots. Okay, there's a series in the plasma membranes. Can you see? Those are the, I mean, that is a set of communicating junctions. Okay. So write down under gap junctions or communicating junctions. There are cytoplasmic channels composed of special membrane proteins. 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 Okay. Next point, forms a pore, forms a pore through which ions, sugars and amino acids can pass. Forms a pore through which ions, sugars and amino acids can pass. Okay. Write down the examples: heart muscle, animal embryo. Heart muscle and animal embryo. Okay, clear. Then earlier when we were talking about cell wall also, I ignored this. There's a small section that I want to teach that is called plasmodesmata. What are the plasmodesmata? Right? Shall I erase this? Clear, right? Plasmodesmata. Okay. So plasmodesmata are not strictly cell junctions. Okay. they are sort of just holes, right? කෙලින්ම අල්ලපු ගෙදර ඔයගොල්ලෝගේ තාප්පේ හදපු හිලක් වගේ. Right? There's no regulation. Anybody can go and come and go. Right? Not like a communicating junction. That's what I want to understand. Right? They are not like communicating junctions. So it's like this. So if you have two plant cells like this, if you have two plant cells like this, okay, I'll make it a bit bigger. Okay, if you have two plant cells like this, a plasmodesmata, okay, a plasmodesmata is a gap in the cell wall. So this is the cell wall of this cell, and it comes to sort of the center, and okay, so this is how the cell wall, the plant cells are separated by the cell wall. Right? This is how the plant cells are separated. If you are said to have a plasmodesmata, that means there's a gap or a hole, right, in the cell wall. Then what about whatever the inner structures? They are separate, right? So this cell has its own vacuole. This has its own vacuole. This has its own nucleus with DNA inside. This has its own nucleus with DNA inside. It has mitochondria. This has mitochondria. Everything all good. What about the plasma membrane? What do you think? So there's an opening in the cell wall. Okay. So what are we talking about here? We're talking about plasmodesmata. Right? Okay. So what about the plasma membrane or the cell membrane? It is going to be shared, right? The plasma membrane. So this is the plasma membrane. It's going to go into the other cell. It's going to go around, and it is going to jump back through this opening, and it's going to come like this. Okay. So the plasma membrane is going to go through this plasmodesmata to surround both cells. Got it? So how do you describe a plasmodesmata? How do you describe a plasmodesmata? They are membrane lined, right? It is lined by the plasma membrane. Got it? Come to the resource book again. Come to the resource book. Over again, plasmodesmata, microscopic channels which runs through the plant cell walls. They are cytoplasmic living connections, right? They are cytoplasmic living connections. Okay, cytoplasmic true, ne? So they are filled with cytoplasm. Then may chuti gap එක through. Cytoplasm is shared, right? So this orange color, if this orange color is the cytoplasm, okay, it is filled with cytoplasm through the plasmodesmata. Got it? So cytoplasmic, next point, living, okay, living. Again. These are membrane lined channels filled with cytoplasm. They are membrane lined. Membrane lined channels filled with cytoplasm. Right? So this red color, that is the plasma membrane, is going to line the plasmodesmata as well. Okay? Clear? Right? Then I said I'll explain to you what is this apoplast pathway. Apoplastic pathway I'll explain. What do you mean by apoplastic pathway? In order to explain the apoplastic pathway, I have to draw another plant cell. Right? So your plants have a very efficient way of transporting water from one cell to the other. Right? You know, your plants, they mainly rely on water based transport, then they don't have a blood, right? So they need to transport water and other dissolved minerals very fast from one cell to the other. Okay, so you have a solution here. Maybe you make a plasmodesmata from one cell to the other. It's a very good connection. It's a direct cytoplasmic connection. Taggala Deval, you can exchange. So this plasmodesmata is the main part of this apoplastic pathway. So the methanoning, if I draw another plant cell, if I draw another plant cell, and if I draw also another one, right? What is the best way to send things from one cell to the other? Is to make a plasmodesmata. So methanol will have a plasmodesmata. Okay. Then if there's another plant cell, right? If there's another plant cell that is starting from here, okay, you can connect that plant cell with this plant cell by another plasmodesmata. Got it? So the apoplastic pathway is basically the transport of material from one cell to the other via plasmodesmata, right? So imagine if it's water from this cell to this through the plasmodesmata, you can transport. Okay, clear. So plasmodesmata is involved in that sort of transport as well. Got it? So no need to write down anything about that right now. I just wanted to show how all the plasmodesmata get together for transport. Okay, clear. Any questions? Right. Then we'll go and do some questions. Harid, come to the question tute. Come to the question tute. So for anyone who's brand new, so this is our class structure, right? We have the main guideline as the resource book, right? Resource book is your ultimate guide. I'm just there to help you understand what is there and to help you organize what is there and also to help you memorize what is there. ඔය තුන තමයි මම කරන්නේ. Okay. So for that we make small notes in the diagram tube. Then we go and do questions in the question tube, and we try to write essays almost every class. Right? That's happening in a different separate essay writing book. Right? Okay. So in the question tube, can we do an MCQ? Yes, question number 13 try කරන්න ටග්ගල්. Page number 3, question number 13. ටග්ගල් try කරන්න. Question number 13. Which of the following statements regarding cell junctions are correct? Which of the following statements regarding cell junctions are correct? It's an ABCD type question. Okay, 2 minutes. ටග්ගල් කරන්න. Online people, send me the answer to the chat. I think you don't need to memorize this. You know the combinations of answers, right? If it's A, B, D, it's 1. If it's A, C, D, answer number 2. Then if it's A, B, 3, C, D, 4, any other combination, answer number 5. Okay? I think you need to memorize. It is given on the top of the before you start the questions, right? Right. When you get closer to the exam, make a matakitno. Okay, these combinations matakitno. I used to remember, but now I can't. ABD is 1, ACD is 2, homo matakitno. Okay. Any other response that is not one of these combinations is 5. Again, one ABC, CD, E. Right? Just one answer. Whatever other than these is 5. Okay. So I think I told you this. Question number 40 to 50. right? Listen, question number 40 to 50, that is the biggest challenge. If you start to get like 555 out, you start to doubt your other answers as well. For the answers you got maybe A, B, C, D, තව හරි වගේ. Or one of the answers is wrong වගේ. Okay? So you think okay, ආපහු මේකටත් 5 ගහන්න බෑ වගේ. එතකොට like so this sort of a mind games this 40 to 50 is the place where even the best students lose at least 1 or 2 marks. මේ සෙට් එකේ ඉඳ තමයි 50 out of 50 නැත්තේ right. Okay, even the marking panel gets sometimes confused. Okay, is it A, B, C, or is it answer number 5, right? So that's why for the bio MCQ that's 50 out of 50 is not there. 49, 48. Got it? Done. What's the answer? What's the answer? Yeah. What's the answer? Which of the following statements regarding cell junctions is are correct? A. Animal embryos have cell junctions which allow the passage of ions. Yeah, no? Israelian, what is the cell junction do they have? Right? So that's the next thing. We use this as another way to learn. We write reviews in front of the MCQs, okay? Product space madhi way, but try to manage it. Animal embryos have cell junctions. What is the cell junction found in animal embryo? Board dege thiyero. Gap junctions or communicating junctions. Right in front. Other cell junctions කියන එකට arrow එකක් දාලා gap junctions or communicating junctions. Next, tight junctions connect the plasma membranes of adjacent cells, forming a continuous seal which prevents leakage of extracellular fluid. Seems good. Yeah, that's a correct answer. Yeah, tight junctions connect plasma membranes, forming a continuous seal. I told you, මේ keywords ඕනේ no prevents leakage, continuous seal. ඔම තමයි මතක තියාගන්න. Okay. C. Plasmodesmata are non-living connections between cell walls of adjoining plant cells. Living. Why do you say it's living? It is filled with cytoplasm and also membrane line. Membrane line. D. Desmosomes allow exchange of signals and materials? No. Desmosomes are mainly concerned about strength. Okay. Then E. Gap junctions attach cytoskeletons of adjoining cells by intermediate filaments? False. You can cut the word gap junction by a different color pen, right? So you can correct it. It's a wrong statement. Who attaches cytoskeletons by intermediate filaments? කවුද? Desmosomes. So anchor junctions. So දෙක මතක තියාගන්න සමහරවිට anchor කියන්නේ communicating the gap කියන්නේ desmosome ද ඔහොම මතක වෙනවා. Okay? Right? Right? So දෙකම මතක තියාගන්න. So in the paper they are going to ask either gap or communicating or desmosome or anchor. දෙකෙන් එකක් මතක තියාගන්න බෑ. Okay? දෙකම මතක තියෙන්න. Got it? So A is correct, B is correct. That's all, right? A and B. Answer number 3. Okay. Okay. 11. Tagga 11 Karan. Question number 11. Question number 11. What's the page? Page number 3. Question number 11. Which of the following is the best combination of cell junction, location and function? Cell junction, location and function. Cell junction, location and function. Modican. That's the answer. Yes. Madi are the answers, Right. Got it. What's the answer? Yeah. Third one. Yeah. This side. Modican. Three. Okay. I don't know. Right. Let's see. Best combination of cell junction, location, function. Tight junction, gut epithelium, communication. What do you think? Tight junctions? Yes. You have in the gut. But is it for communication? No. එතන අපි කොහොමද ඔක හදන්නේ, right? You can, I think, if things, since two things are correct, you can cut communication and write there prevents leakage. හැබැයි කපද්දි different color, red or pink හරි මොකක් හරි sort of not black or blue, okay, right? Because we are correcting, not correcting the question, we are sort of writing your own review. නේද? Tight junction, gut epithelium, what is the proper function? Prevents leakage. නේද? So bio class එනකොට පාට පෑන් ටිකක් ගේන්න. හරි? Physics class යනකොට එක පෑනක් ගෙනියන්න. That's enough. Chemistry යනකොට එක තව පැන්සලක් ගෙනියන්න. That's enough. Bio එනකොට bring your maybe your sisters, boys cover a sister or someone's pencil case at least. I used to do that. Right? මම bio class යනකොට I used to take my sisters highlighters. Right? To anchor junction, skin epithelium prevents leakage. Skin epithelium has to prevent leakage antima deka hari ne, that is the location and function, okay, but what is the proper cell junction? Yeah, tight. Tight. So cut the anchor and correct it as tight. Okay. 3, tight junction gut epithelium prevents leakage. Yes, very good. That's the correct answer. Gut epithelium. 4, gap junction nervous tissue prevents leakage. Actually, so gap junctions are found in nervous tissue. Okay, but what's the wrong thing? Prevents leakage is not a proper function. Right? Gap junctions are found in nervous tissue, even though it's not given, because this is before you are sort of the revision of the new syllabus. So some things have been removed. Okay. So because of preventing leakage, it's false. Then 5, anchor junction skin communication. Nothing is matching. Okay, because anchor, what it does is prevents leakage. Skin epithelium has tight. Communication is by gap. Ne? ඔය යටින් තුනටම ලියා ගන්න ඕන, right? So skin epithelium එකට ලියා ගන්න, what do you have? You have a tight junction. Under communication, write down, it's a gap junction or communication. Got it? Okay. Right. Then, if you come to page number 4, next page. There's a structured essay. Okay. There's a set of structured essay questions. We didn't do any of that, right? We didn't do any of that. Okay. You can do everything. 1A, what is a cell junction? B, name a cell junction carries out each of the following. Then name the structure that helps in cytoplasmic streaming. Okay, that is from cytoskeleton. State the location of the secondary cell wall. 3 functions of rough endoplasmic reticulum. 3 types of vacuoles. You can refer the resource. පොත් බලන්න අවසර තියෙනවා. What's the issue? In the 4th one, gap junction, nervous tissue preventing leakage, there's no need to write anything there. Okay. Do the structure essay question. Uh, so actually, for a structured essay question, kochara time hambenada? 15 minutes. But is that a complete structured essay question? No. That is only like a structured essay question has part A, B, C. ඔතන ඔය එක part A වගේ ඔච්චරයි. That is one third of a structured essay. Okay? So එතකොට හරිනම් කොච්චරද time එක දෙන්න ඕනේ? Only 5 minutes. But okay, take around maybe 8, 10 හම්බෙනවා I think, right? Okay. Well, people found out the place. Page number 4, I think. The question tutor is available in the Zoom chat as well. Tak galakan. You can refer the resource. But my point is, whenever you are writing the answer, try to close the resource and write it by yourself. පොඩක් refer කරන්න ලියන්න. නැත්නම් මේක පොඩි කාලේ වගේ it's not copywriting, okay? Every row has a resource book, right? Yeah. I don't forget that the bio right? So get your own personal resource book at this point, okay? I'm not selling resource books, so I can't provide you one. Every row has one, right? Can you share it? Okay, still writing? Online people, how is it looking? Still writing? Right, shall we discuss? Anyone who's still writing? First question, what is a cell junction? What is a cell junction? Mammalian, na? What is a cell junction? We have to sort of formulate a definition using combining two sentences. For cell junctions, we can write an answer like this: a structure at which cytoplasms of adjoining cells are joined. A structure at which cytoplasms of adjoining cells are joined to interact and communicate are joined to interact and communicate to interact and communicate via sites of direct physical contact. Interact and communicate via sites of direct physical contact. Via sites of direct physical contact. Got it? Next, name a cell junction that carries out each of the following: preventing leakage of extracellular fluid, tight, tight junction. Okay, tight junction. Allowing exchange of material, gap junction, communicating junction, gap or communicating junction. Okay. 2. Name the structure that helps in cytoplasmic streaming, movement of chromosomes, and state its structural components. That is cytoskeleton. No? Cytoskeleton. We talked about this last week. Cytoskeleton. What are its three main structural components? You have microtubules, yes, microfilaments, or you can say actin filaments, then intermediate filaments. Okay. intermediate filaments. State the location of the secondary cell wall of plant cell and name a substance present in it other than cellulose. Hurry. How do you describe the location of origin? Where is it formed? How do you describe? Secondary cell wall. How do you describe? Yes. It lies between plasma membrane and primary cell wall. Okay. Lies between plasma membrane and primary cell wall. Right. So outer to plasma membrane, inner to primary cell. Okay. එම සැන්ඩ්විච් වෙලා තියෙන්නේ between plasma membrane and primary cell. Okay. But realistically, can you have a plasma membrane if you have a secondary cell wall? No. Right? Once you develop a secondary cell wall, the plasma membrane is going to die, ne? Because the cell is going to die. ඇතුළ හිස්. Just the casing. Secondary cell wall and the primary cell wall is going to remain. Right? Cytoplasm, plasma membrane, everything is going to be dead. 4. State 3 functions of rough endoplasmic reticulum. Then අවුරුද්දට විතර කලින් කරේ නේ. RER. What are the functions of rough endoplasmic reticulum? You can write several නේද? RER. What can you write? Transport protein synthesized by bound ribosomes, synthesize glycoproteins, produce transport vesicles, synthesize membrane phospholipids, function as a membrane factory by facilitating the growth of own membrane. Okay, any three you can write. Okay. Transport protein synthesized by brown ribosomes. Any one of those you can write. Okay. Okay. Hi, there's another one. One more. Name 3 types of vacuoles. Who is the big one? Central vacuole. Then who acts like a kidney? Sort of in the water balance, solute balance? Contractile. Then who is your digestive function one? Food vacuole. Central, contractile, food. Vacuoles jati tunai. Okay. We'll talk about these food and contractile when we are talking about protists. Your Euglena, Paramecium, amoebas. When we are talking about those, we'll come across these contractile and food vacuoles. Central vacuoles are mainly found in what? Plant cells. Those we'll talk about in the 4th lesson, plant form and function. Okay, right. Yeah. So for your 2027 batch, there's going to be a term test next week. So that is going to be the first 5-hour paper that they are facing. So until the end of plant form, there are going to be questions. So okay, term test, and for them, for the physical class, I'll be ranking the students. Okay, there's going to be a higher scorer, second and third, and for the top scorer, there's going to be a prize. Right? For online people also, there's going to be 1, 2, and 3, because the papers are going to be marked. Okay, and you're going to be ranked. Online people also, you can send the paper to be marked, and it will be marked and sent back to you as a PDF. Okay, so for you and Egolanta, exactly as an exam setting, I'm going to give the paper as exactly at the time, and after 5 hours, I'm going to collect it. And for online people, I'm delivering the paper, so you have to open the sealed envelope at the exam time. Okay, while the video is on, there's a sort of a program to sort of provide you the exam, let's say, exam experience, not the experience, exam experience kind of there, right? Exam practice mentality. 5 hours, not exactly 5 hours, but 2 hour, 3 hour gaps. That's also takes some practice, right? Because you are not just sitting there, you are sitting there under some tension, right? So sometimes the time may pass very fast, sometimes the time may not pass at all. Okay, so some things you need to practice. What are the things you need to practice? Kondari, that's a problem. Okay, because you are sitting on wooden chairs, because we are facing the exam in Sri Lanka. Even right now you are Okay, but at least it's AC. So in the exam hall, it's not going to be AC. Sometimes if it's raining, paper එකත් එමෙයි window එකෙන් කියන්න බෑ. Depends on where you get the desk. Main problem, toilets. Bladder control, bowel control තමයි main problem. I'm serious. It's a big issue. Okay, very big issue because you are not allowed to go to the washroom. Yes, during the exam, you need to take a chaperone with you. Right, chaperone කියන කවුරු හරි සර් කෙනෙක් හරි you have to, they have to come with you to the washroom. Someone from the exam staff. Okay, so if you can't control for 3 hours, that's going to be a problem. I had to go to the bathroom in the middle of the physics paper, second paper. So I had to wait until they send someone. තනිය යන දෙන්නේ. Okay, so that's something you have to practice. Sometimes you can't help it. Okay, but if that problem arises, my advice is get rid of it right there because it's going to affect your performance. Okay, because some of your brain function capacity is going to be directed there. Okay, I need to go to the washroom, right? So if you really have to go, you have to go. Okay, but try to control it. Try to control your water intake. ඒ මයිකල් වතුර නොබී ඉන්න එපා, dehydrate වෙලා, right? When you are dehydrated, the brain doesn't function as well, right? So you have to balance both, right? It's a big problem. Okay, right? I'm serious, right? So anyway, we'll move on to the next topic. That is the final part of cellular structure, uh, extracellular matrix. Okay, any questions, online people, from the things that we discussed? Okay, extracellular matrix. This can be a very nice short note question. Okay, that's going to be our next homework. Short note from extracellular matrix. Okay, so if you take your plant cells, they are all interconnected by, sorry, they are surrounded by cell walls. Then cell walls are glued together by what? Cell walls are pasted together by what? Pectin. Pectin. Right? But if you take your animal cells, how are they? I mean, they are only surrounded by the plasma membrane. No? Plasma membrane is a very fluid, right? There's no strength to it, right? It's just a barrier. So how are your animal cells maintaining their shape? How are they supported? That is done by your extracellular matrix, right? Particularly in the type of tissue that we call as connective tissue, that is sort of found underneath your skin, okay? That is attaching your skin to the muscle underneath, okay? Then holding your organs in place, right? Around the liver, around the heart, everywhere you have connective tissue. In sort of the cells you find in connective tissue, you have a very nice supportive structure around those cells that we call as ECM, extracellular matrix. Okay, so that's what we are going to talk about next. So again, the extracellular matrix is not a very what we say, the structure is not a uniform, I mean universal structure. It can change from tissue to tissue. Okay, but here we are going to study like a common model, like a model of the extracellular matrix of how the most, let's say, the common form of the extracellular matrix that you can find. Okay, let's read what is given in the resource. Product confusing, but I'll explain. ECM, page number 39. Okay, read. Although animal cells lack cell walls, they do have elaborate extracellular matrix, ECM. Main components of ECM are glycoproteins and other carbohydrate containing molecules secreted by the cells. The make you open up your diagram tube, page number 7. I have given a full color image of the ECM that we are talking about. Page number 7. Okay. It's very elaborate. Right? Elaborate means complicated. Okay. Most abundant glycoprotein in the ECM is collagen. Can you highlight that? It's not a definition but a theory point. Huh? Most abundant glycoprotein is collagen. Okay, which forms strong fibers outside the cell. Collagen fibers are embedded in a network woven out of proteoglycans secreted by cells. Collagen fibers are connected to integral proteins of plasma membrane via fibronectin. Alright, over again. So, in order to draw your extracellular matrix, I need to draw your plasma membrane as well, right? So, your plasma membrane is like what? It's a phospholipid bilayer, right? So, how do you draw phospholipids? By its symbol, it has two hydrocarbon chains which are hydrophobic, right? And it has a hydrophilic head. Okay, so this is your phospholipid bilayer of the plasma membrane, right? So here I will draw a transmembrane protein, that is going the entire thickness, right? A transmembrane protein is there. Matakai the different types of integral proteins. Okay, so this is your plasma membrane. No? So how is your extracellular matrix arranged? It is mainly composed of collagen in the model that we are studying. Okay, it is mainly composed of collagen. Collagen, among, if I ask you what is the level of organization in collagen, yes, level of protein organization in collagen. One of the levels are there: primary, tertiary, sorry, secondary, tertiary, and quaternary. Ne? The collagen is a what level? Quaternary. It has three polypeptide chains. Ne? Matakai? Right. Matakai, yes. Okay. So collagen is a thick protein fiber. Collagen is a thick protein fiber. So it forms these sort of fibers around the, sorry, outside the cell, right? These are all collagen fibers that are part of the extracellular matrix. Okay, they are sort of, they are to provide strength. Madagaina collagen provides mechanical support. Okay, these are the collagen fibers. Okay, then look at the next sentence, which collagen fibers form strong fibers. They are embedded in a network woven out of proteoglycan. Proteoglycan. What do you mean by proteoglycan? I have given the structure of a proteoglycan molecule here. Modigan. It's like a, have you seen our hydrilla plants in a ponsal hadina? Right? This sort of aquatic plant. And a wage the structure, okay? So you have a central core of polysaccharide. We have a central core of polysaccharide, you know. From there you have this branching protein branches, okay? These are sort of protein branches, and from each you have again sort of polysaccharide pieces that are coming out like this, okay? This is what we call as a proteoglycan complex. The main structure you don't need, okay? Can you see there's a polysaccharide molecule and there's a core protein, then you have proteoglycan sort of, sorry, carbohydrate branches. Then we make it a, this is a single proteoglycan molecule. मैं own है, okay? This is a single proteoglycan molecule. This entire thing is what we call as a cat. proteoglycan complex. Okay. So proteoglycan complex. So I'm going to draw this entire proteoglycan in this sort of green color, right? These proteoglycans are sort of going through your collagens, right? So your collagens are sort of embedded or immersed in this sort of proteoglycan network. Okay, your collagens are immersed in this proteoglycan network. Okay, so the composition of this proteoglycan can change from different tissue to different tissue. Okay, so many different types of proteoglycans are there. Okay, depending on the tissue, there are different types of connective tissue, dense connective tissue, loose connective, your cartilage is a connective tissue, your bone is a connective tissue. Okay, so depending on those tissue type, this proteoglycan can change, but definitely you have a lot of collagen. That collagen is embedded in this proteoglycan. There, right? Then next sentence, what do you say there? What do they say? Collagen fibers are connected to integral proteins of plasma membrane via fibronectin. No. ये मुकद्दर देखिए. Okay, so they have collagen fibers that are going like this. Right? These are all collagen. You need to remember your extracellular matrix provides strength to the plasma membrane as well. How does it do that? That happens via a communication or a contact between your extracellular matrix and your inner supportive structure. What is your inner supportive structure? Cytoskeleton, right? So you have cytoskeletal fibers such as your microtubules, actins, okay, intermediate filaments, okay. There's like cytoskeleton is there. No? This cytoskeleton is going to be connected to certain proteins of the plasma membrane. Okay. Then, so it is connected to this protein. Then your extracellular matrix is connected to that protein via a special protein called What is the name of that protein? Okay. Fibronectin. Oh. This is your outer supportive structure. This is your inner supportive structure. They are connected. Alienut supported denu, Ethylene supported. Okay. Let's label this. So the blue color is collagen. This is plasma membrane. That is fibronectin. So this green color I'll just label as proteoglycan. Proteoglycan. This is, we'll say this is actually a transmembrane protein, but because it's given in your resource as a integral protein. Yes, I know transmembrane proteins are a type of integral protein. Integral protein means what? Any protein that is sort of in the plasma membrane, inside. Yeah. So this is actually a special type of integral protein that is called as a transmembrane protein. So anyway, we'll call it an integral protein. That's it. Okay. So the proper diagram is given in your tute. This is a simplified diagram. Okay. Collagen fibers I have given in purple color. Fibronectin, can you see? Proteoglycan complex. Then integrins keladine. Okay. Integral proteins in your syllabus. Okay. Right. What are their functions? Read in the resource. Read. Forms a protective layer over the cell surface, linking the extracellular matrix and cytoskeleton. Highlight those. You can see, right? ECM and cytoskeleton is connected. Yeah. Then influences cell behavior by involving in mechanical and chemical signaling, right? So this connection between ECM and cytoskeleton helps to control the cell and also to coordinate the functions of the tissue, how it is going to behave. Imagine this is part of a cartilage, right? If the cartilage is going to become more stiff, or if it's going to repair themselves, or if it's going to grow bigger, whatever the tissue is doing, it is going to be coordinated nicely because of the ECM. The may ECM mega is going to be shared into the next cell. Yeah, there's another cell next, right? It is shared, the ECM. Okay, that's how a multicellular organism's tissue functions. All the cells are nicely connected. Okay, right. So come to the diagram tute, page number 7. ECM is there. Let's write down some points. What points take ලියමු dots තියලා. Okay. First point, composed of, right, write down. First point, composed of මොනවාද? Glycoproteins. First one, composed of glycoproteins and other carbohydrate containing compounds secreted by cells. Okay. Carbohydrate containing molecules කියලා ලියනවා. Right. හරිද? Then next point, star mark එකක් දාලා ලියා ගන්න. Okay. Composed of glycoproteins and other carbohydrate containing molecules produced by secreted by cells. Next point, write down. Okay. Most abundant glycoprotein is collagen. Most abundant glycoprotein is collagen. Okay. Next point, another normal point. Collagen is embedded in a network of proteoglycan. 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 තව ඉඩ තියෙනවනේ? තව ඉඩ තියෙනවද? Okay. Final point. Collagen fibers are connected to integral proteins. Collagen fibers are connected to integral proteins. Collagen fibers are connected to integral proteins. Collagen fibers are connected to integral proteins by fibronectin. Integral proteins by fibronectin. Right? එච්චරයි. ECM. දැන් මොකද කරන්න අපි? Any MCQ that we haven't done. මොහරි කරපු නැති එකක් තියෙනවා. Any MCQ we haven't done. හොය කරමු. There's one more I think we need to do. Any questions? What are the questions we haven't done? මොනවද අපි කළේ නැත්තේ? ටක් ගාලා කියන්න. 1 yes. Yes or no? First question, page number 1 ඒ කියන්නේ මේ cellular structure කියන එකේ question number 1 have we done? Yes, we have done. 2 have we done? 2 no. Yes or no? Okay. 2 කළා නැහැ. හරි. Right. දැන් මොකද කරන්නේ? අපි ඒ ටික කරන්නේ නැහැ. Right? So 2 කළා නැහැ. Then 3 have we done? 3 තාම කරන්න බෑ. පොඩ ඉන්න. 4 have we done? 4 කළා නැහැ. First එක කළත් නැහැ. Okay. Very good. 4 කළා. කළා නැහැ. කළා නැහැ. Okay. 5 කළා නැහැ. මොකද? Right? 6 කරා. 7 කරා. 8 කරේ නැහැ. Okay. 9 කරේ නැහැ. 10 කරා. 11 දැන් කරා. 12 කරේ නැහැ. 13 කරා මේ දැන්. 14 කරා. 15 කරා. 15 කරා. Okay. අචුට්ටයි. හරි. Right. Okay. So we are going to come to the final part of this lesson. Okay. මොකද්ද? මෙන්න මේක. මේ බෑක් කවර් එකේ තියෙන chart එක. ඔන්න ඕක කරන්නේ. 7 කළාද? Okay. There's this comparison chart නේද? What is that? The comparison between prokaryote and eukaryotic cells. I told you I'm going to do this at the end of discussing all the cellular structures. දැන් ඔක්කොම දන්නවා. Starting from the nucleus, starting from plasma membrane till the extracellular matrix, you know everything that you need to know as a A-level biology student. And with that knowledge, we are going to do a comparison between eukaryotic versus prokaryotic cells. So in your resource, that is in page number 25 and 26, neda? 25 and 26. Very important comparison. Okay. Turn to that page, page number 25 again. Online people, resource, page number 25. In your syllabus, there are these sort of මහා එපා කරපු charts ටිකක් තියෙනවා, right? Okay. There's this chart. And in your evolution and diversity, there's a comparison between bacteria, archaea and eukarya again. And in plant form, there's a plant vitamin chart. Plant mineral chart. Then in your human biology, there's a vitamin list එකක් තියෙනවා. ඔලවලට තිබ්බ නේද? මලාට මතක හිටින්නේ නැහැ නේද? Vitamins and functions, deficiency symptoms. Okay. In A levels, I will teach them with some nice diagrams so you will remember. හරිද? beriberi හැදුනාම මොනවද වෙන්නේ? පැලිග්‍රහ හැදුනාම මොනවද වෙන්නේ? Okay. Have you heard these diseases? Vitamin deficiency. Scurvy හැදුනාම මොකක්ද වෙන්නේ? I will show you and explain. හරිද? Anyway, there are set of charts like this. So those charts you have to study very frequently. තේරුණාද? Those kind of charts you have to study very frequently. Otherwise මොකද වෙන්නේ? අමතක. And the other thing is you cannot try to memorize them even at one sitting. නිකන් අද මේ chart එක පාඩම් කරලා තමයි නැගිටින්නේ කියලා, that's not going to work. Okay? That's because they are very, they have a lot of theory points and they are very boring to study. මතක හිටින්නේ නැහැ. එපා. So what I recommend is you study them frequently, at least once a month. Study. Okay, that's why I gave it as the cover of the tute as well. තව දැන් මාසයක් විතර we are going to do this tute එතකන්වත් මේක පොඩ නිකන් මං එනකන් හරි පොඩ්ඩක් try to remember one of the theory points given here. Okay, right, come to the resource. We are going to do a comparison between prokaryotes versus eukaryotes. Okay, we are going to do a comparison between prokaryote versus eukaryote. Then some examples for prokaryotes are bacteria and archaebacteria. Bacteria and archaebacteria are your main prokaryote groups. Okay, so bacteria are the bacteria that you find usually everywhere, on your mouth, on your skin surface, on your desk, everywhere, on your phone, on tarantira, right? Those are the common bacteria. Archaebacteria are sort of special bacteria that are very resistant, very hard. They are found in these volcanoes, hot water springs, sort of very salty, sort of deep sea under very pressure, very cold environments. You can find these archaebacteria. Jump to eukaryotes. What do you have? Protists, fungi, plants, animals. Right. Then cell size. Highlight prokaryotes 0.5 to 5 micrometers. Okay, 0.5 to 5 micrometers. Then eukaryotes 10 micrometers to 100 micrometers. 10 micrometers to 100 micrometers. You can see sort of larger in size. Okay. Prokaryotes are mainly unicellular. Okay, prokaryotes are mainly unicellular because this kind of some kind of cyanobacteria can form colonies, right? Bacterial cells can join together to form a colony. So that kind of looks like a multicellular structure, but it's not. Okay, they are mostly unicellular. Maybe at the eukaryotes mainly multicellular, right? Except most of protista and some fungi. If you take your euglena, paramecium, amoeba, those are eukaryotes, but they exist as single cells. Okay. Then evolutionary origin 3.5 prokaryotes, 1.8 highlight. Theory points, okay. Then cell division of prokaryotes, it's binary fission. Binary fission කියලා කියන්නේ, that's something we'll talk about when we are doing cell division. So, okay. So imagine you have a bacterial cell like this. Okay. In that bacterial cell, there is no nucleus. Okay. There's no nucleus in bacterial cell. I think I showed you with the software, right? When? I'll show you today. Right? There is no nucleus in bacteria. Their DNA is found just freely in the cytoplasm. Okay, just freely, no nucleus. So when this bacteria thinks it wants to divide, what's going to happen is there's no proper cell division. Mitosis වගේ එකක් වෙන්නේ නැහැ, right? What's going to happen is it's just going to divide, and නිකන් by chance one of the daughter bacteria is going to get some of the DNA and the other daughter is going to get some of the DNA. It's not equally divided. Just some parts are going to go here and there. So it's just going to divide like this. Binary fission. Binary means what? Dekai. 2. Fission means breaking apart. Binary fission. Okay? That's how you understand. Binary fission is breaking into 2. Okay? That's why bacteria are so dangerous. There's no proper regulation. Right? If one of your cells are going to divide, there's a nice procedure. So today we are going to learn. Before a cell even divides, there's a internal quality control. There are some molecules in your body that's going to come to the cell and say, okay, this cell is not ready to divide right now. Cell division. It's not properly prepared. How did a DNA copy? Right? Not properly prepared. So if one of your cells is not qualified to divide, it's going to stop it. Divide වෙන්න දෙන්නේ නැහැ. අර license එක cancel fail වෙනවා වගේ trial පෙන්නන්න ගිහලා license හම්බෙන්නේ ඒ වගේ your cells will not be allowed to divide. But bacteria cells are not like that. can divide anytime. So that's why they change, they mutate. So many different bacterial variations arise. Okay, that's why antibiotics stop working. Okay, that's one of the big problems. I won't stop today. Okay, antibiotic resistance because of the bacteria can change very fast. Other thing antibiotic you can't use tomorrow because the bacteria has become resistant. Got it? Cell division of eukaryotes, mitosis, meiosis, or both. Okay, proper cell division is there. Genetic material DNA is circular and lies free in the cytoplasm. This region is called the nucleoid. Highlight. I'll show you the nucleoid. Circular DNA is found in the nucleoid region. DNA is not associated with histone proteins. DNA is not associated with histone protein. What do you mean by histone protein? Shall I erase this proteoglycan? What do you mean by histone protein? I think I explained this when we are talking about the nucleus. Yeah? So today in cell division also we are going to talk about that. If you take your DNA, what do you mean by your DNA? Eukaryotic DNA. Your DNA is tightly and very nicely packaged. So if you take, if this is your DNA double helix, we draw it like a single DNA strand, right? This is your DNA double helix. We organize it in your cells by wrapping it around some spherical proteins, bola proteins. These proteins are called histones. So when you wrap this DNA around this histone two times, then again this DNA fiber is going to go, and then you again wrap it around some more histones. You pack this. You pack this DNA very tightly because you have so much DNA. A eukaryotic cell has so much DNA. Nucleus you have to somehow pack. Yeah. So this level is what we call as chromatin. This is what we call as chromatin. So there is no such chromatin in your prokaryotic cells. Oh, hey, DNA. Got it? Then what's going to happen is this chromatin, whenever your cell is about to divide, it is going to pack even further. So this level, it is going to, okay, it is going to sort of coil themselves like this. Then that coil itself is going to fold like this to form a very compact structure that we call as chromosome. Chromosomes are found only when the cell is about to divide. Okay. Nathan normally chromatin vidiyata the DNA is found in eukaryotic cells. Okay. So on the eukaryotic side, read, okay, DNA is linear, containing nucleus associated with histone. These are histone proteins. Okay. Did you highlight nucleoid found in the nucleoid region? Okay. Then type of ribosome, prokaryotes have only 70s. Highlight. I told you the May chart, if I try to teach you this chart before I taught you about ribosomes, because you don't know what is 70s or 80s. But now you know. On the eukaryotic side, both 70s and 80s ribosomes are present. So you know where are the places you can find 70s in mitochondria and chloroplast. Where do you have 80s? In two forms. You have bound or free. Bound is bound to rough endoplasmic reticulum. Free is just found in the cytosol as groups. Next page. Right? Organelles. No membrane bound organelles are present. Okay? There are no inner membranes. Right? Consist of few subcellular components. No organelles. Can you highlight? No membrane bound organelles are present. Prokaryotes are not. Consist of few subcellular components such as ribosomes. Subcellular components are mainly ribosomes. Internal membranes are scarce. If present, are usually associated with respiration, photosynthesis, or nitrogen fixation. Those are the main things done by the inner membranes of prokaryotes. Jump to the eukaryote side. Manatin, membrane bound organelles and subcellular components are there. There are two double membrane bound organelles, then there are single membrane bound organelles. So, if you take a eukaryotic cell, what are the double membrane bound? Yeah, where do you have double membrane? Nucleus, yeah, chloroplast, mitochondria. Right? What are your single membrane bound? You have vacuoles, lysosomes, Golgi, yeah, those kind of things. Cell walls. Cell wall of prokaryotes, peptidoglycan in bacteria and cyanobacteria, polysaccharide and protein in archaebacteria. Highlight that. Peptidoglycan in bacteria and cyanobacteria, polysaccharides and proteins in archaebacteria. Cell walls on the eukaryotic side, cell walls of green plants and fungi are rigid and contain polysaccharides. You have cellulose in plant cell walls and chitin in fungal cell walls. Those are the two cell wall types. Then there's another important comparison, the structure of the flagella. Okay, so we have already talked about flagella. Okay, on the prokaryotic flagellum, it's simple. The structure is very simple, lacks microtubules. It is just composed of simple fibrous proteins. Microtubules will never composed. It is extracellular. Highlight that. Prokaryotic flagellum is extracellular. Very, very important. Not enclosed by a cell surface membrane. 20 nanometer. They can ask you what is the average size of a prokaryotic flagellum? 20 nanometers. Then jump to the eukaryotic side. This is the flagellum we learned about. That is composed of microtubules with what? 9 plus 2. Ne? What do you mean by 9 plus 2? There's 9 doublets on the outside and 2 central single microtubules. Ne? 9 plus 2 arrangement of microtubules in the flagella, and this whole thing is surrounded by the plasma membrane. No? So it is intracellular. Size 200 nanometers average. Okay, if you take one of your sperm cells, yes, that is the size of the flagella. Yeah. Finally, last set, respiration by internal membrane foldings. You don't have specialized structures. If you come to eukaryotic, you have mitochondria. Photosynthesis, prokaryotes do not have chloroplasts. Takes place on membranes which show no stacking, right? No complicated thylakoid membrane arrangement. You have chloroplasts, contain membranes which are usually stacked into lamellae or grana. Then I hope you can remember the structure of a thylakoid, how the thylakoid membranes form these sort of stacks. That are called grana. Yes, they are found in only in chloroplast. Okay, you can't find those in any of the prokaryotic cells. Nitrogen fixation, some have the ability, none of the eukaryotic cells have the ability. So we have to rely on bacteria for our nitrogen requirement. Okay, we obtain nitrogen from where? From the food we are eating. How did the food get that? For example, if you are eating beef or chicken, how did they get from a plant matter. No? So plant, how did they get the nitrogen? From soil. Where did the soil get the nitrogen? From bacteria. Where did the bacteria get the nitrogen? From the atmosphere. So can you appreciate this? Atmosphere ke nitrogen. Have you done structure and bonding? Chemistry. Still in atomic structure. Okay. Didn't go to the second lesson, right? So in structure and bonding, you will draw the molecule, the shape of the N2 molecule. Name it. There's two lone pairs, I think, right? Connected by a triple bond. This is how nitrogen is found in your atmosphere. 70% of the atmosphere is nitrogen, but it is useless. We can't extract. Because why? This is a very strong bond. Triple bond. Make kadanne. To break this, you need special enzymes. Those enzymes are only found in certain nitrogen fixing bacteria. Nitrosomonas, nitrobacter, only they have the ability. They can convert this N2 into forms that we can use, such as I write here. What are the nitrogen forms that you can use? Nitrate, NO3 minus, or ammonium, NH4 plus. Those are the usable forms for plants. Okay. So without your nitrogen fixing bacteria, we cannot survive. Got it? Very important point. Okay. Then in the question tute, mark these questions. As homework, please do all these questions. Okay. Mark these questions and do all these questions as homework. 1, 2, 3, 4, 5, 8, 9, 12. Okay. We'll check next week. Online people, these MCQs are homework. Okay. 1, 2, 3, 4, 6, 9 homeworks. Sorry, 9 MCQs. 10-minute pedak. 10-minute podi pedak. But I recommend put a padang karla try again, right? That is going to sort of give you some additional benefit of doing the questions. These MCQs are your homework. Okay, right. So that's it. We'll take a small break, and after the break, it is cell division. Harid, any questions? No. Yes. Anything? Okay. Online people, any questions? So for online people, my advice is don't make it a habit of watching recordings. Okay, if you are unable to join live, that's fine. I mean, due to a valid reason, but I prefer if you join with the live until the end. Okay, don't make it a habit because I'm not underestimating you, but at this point you don't have the sort of the concentration ability to sort of watch a recording completely. Okay, there. Stress for a 2026 student maybe okay because they are stressed they will watch a recording completely 1.5 hari 2 times hari dala hari balno. I don't think at your stage you have the concentration sort of or the motivation to watch a recording completely. Okay, so try to join with the live the entire duration. Anyway, small break. 15 minutes. Any question? Okay, right. Everyone's here. Yes. And next, so these questions are for homework, right? So we'll check them first thing next week. Okay. So shall we move on to cell division then? Yeah. That's I mean sort of the today's. I was planning to start it a bit earlier, but that's fine. Okay. So first, I thought of I'll give you a small sort of visualization of different cell divisions that we are going to encounter, because I think in O levels you only learnt about what's going to happen. I mean, what's the basic idea of mitosis? What's the basic idea of meiosis? Yeah, and how many daughter cells are produced? That's the. I mean, you only learned the basic things. I think, right? So in A levels, as of course it's advanced level, right? We are always going to go a bit more ahead with everything. Okay, so that's what I thought. That's why I thought at the end we are going to you are going to be able to understand everything I'm showing, going to be showing right now. But this is like the before the movie starts, you watch a trailer, right? So that's kind of like what's going to happen. Okay, right. So before we move on to that, I think I just wanted to, wanted to show you. Online people, can you see? Yes, you can see, right? So in this bacterial structure, so this is a simple bacterial cell. You can see there's a single flagellum. Okay, and so that simple flagellum is what we call as a extracellular flagellum, because you can see this is the plasma membrane. Okay, now you can see it is slightly highlighted. This is the plasma membrane. So you can see that plasma membrane doesn't extend to cover the flagellum, right? It doesn't extend to cover the flagellum. It is just surrounding only the cell only. So that's why we say it's an extracellular flagellum that you find in prokaryotic cells. What I mainly wanted to show you is this. This is the genetic material or DNA. This is how your DNA is arranged in a prokaryotic cell. There is no nucleus. So you might think, okay, mara DNA godak තියෙනවා. No, it is simply the prokaryotic cell is small and the DNA is not very tightly packed. Okay, therefore, it is sort of almost everywhere in the plasma, sorry, everywhere in the bacterial cell, but we specifically call that region as the nucleoid region. Okay, better. No? Okay, online people better, right? Hurry. Then what else I can show you about the prokaryotic cell? There is we'll talk about these two structures when we are doing molecular biology. Those are called plasmids. Plasmids are pieces of DNA that are not part of the main piece of DNA. That is the DNA found as the nucleoid. Okay, these are called plasmid DNA. Plasmid DNA are sort of important for bacteria for their, what we say as, for their pathogenic activity. So for example, bacteria that are causing certain diseases have their disease causing genes in this plasmid DNA. Okay, so that's why sometimes this plasmid DNA can undergo different changes and make the bacteria more dangerous, right? They are sort of independent pieces of DNA. You don't have those. You have only your DNA that is found in your nucleus. Bacteria, they can find, I mean, they have so many, they have so many plasmids as well as DNA in the nucleoid. Then you can see there are no, usually there are no other organelles inside. Yeah, you can have small, small membrane foldings, But basically that's it. You can see some ribosomes here and there. 70s ribosomes here and there you can find. That's it. Got it? Um, yeah, so I think I showed you a eukaryotic cell last week. No? Okay, so eukaryotic animal cell, you can see so many organelles are there. Double membrane bound organelles are there, surrounded by a plasma membrane. Okay, the DNA is found inside the nucleus. Got it? You can have double membrane bound, single membrane bound. This is such as this. This is a peroxisome. This is, you can have lysosomes here and there, right? Double membrane bound, single membrane bound organelles are there. What I wanted to show you is our cell division. Okay, and yeah, before that, did I show you how DNA packages in your eukaryotic cell? So this is the DNA wrapping around histones. Can you see? That DNA is going to wrap around itself, okay, to become even more tight. Okay, then it is going to coil over itself. It's coiling over itself. So further and further compacting, over compactula, it's going to become finally. This is what is going to form the chromosome, right? So this is like a more complete image. Can you see? This is the DNA double helix stage. It is going to wrap around histone within. This is the histone wrapping around. This stage is called what? Chromatin, right? Chromatin is going to further coil like this. That is going to further coil like this. That is going to, I mean, at the end, you are going to get this very compact structure that we call as chromosome. This is the travel ready DNA, right? Ready to divide. Got it? This understanding is very important because we are going to start next cell division. Before cell division starts, cells are going to produce this packed form of DNA that is called chromosome. Okay. So, what's going to happen in mitosis, right? Before mitosis, right? So, this is a proper mature cell that is about to divide by mitosis. I just show you, right? Did you learn about different stages? In O levels, you didn't have no? Prophase, anaphase, metaphase, over Tibbinen. Okay. Fresh things we are going to learn here. So, this is a cell that is about to be divided by mitosis, right? So, at the end, what is going to be the spoiler alert? You are going to end up with two cells. You are going to end up with two cells. What do you call these two cells? This is the original, this is the mother cell. The mother cell is going to divide into two daughter cells, right? That idea is okay. If a cell divides by mitosis, the two daughters are genetically exactly identical. Both daughters are going to get the same DNA, exactly same DNA, no change. If mitosis happen properly, in proper mitosis, DNA is given exactly to the two daughters, exactly as it is. Okay, that is going to be done by a set of steps. So for simplicity, we are going to get rid of all the other organelles. We are going to get rid of all the other organelles because our main focus is going to be what's going to happen inside the nucleus. So you can see inside the nucleus, you can see the chromatin is going to be condensed into chromosomes. Right. Now you can see chromosomes. Then what happened? Purple. What did that? Shatter What is that? There is a nuclear envelope around the nucleus. Nuclear envelope is going to be removed. With the nuclear envelope, right? The cell is about to divide. So you can see the nuclear envelope is going to it is going to disappear. Who is this? There is a nucleolus. Can you see? Nucleolus is going to disappear as well. Make out the main. Those are the apparatus or the sort of the operator of cell division. That is what? The centrosomes. Centrosomes. Matakai? Centrosomes, there's going to be two centrosomes. The centrosomes are going to gradually come to the two opposite ends. Then they are going to be attached to the chromosomes. They are going to get attached to the chromosomes, and the chromosomes, in this stage we have of a single chromosome we have two copies. Copies දෙකක් තියෙනවා. ඇයි? Daughters දෙන්නෙක් ඉන්නවා. For the two daughter cells, the mother makes two copies of DNA. Right? The mother makes two copies of DNA to give to the two daughters. Yes? Yeah? What's this centrosome and this whole, can you see yellow color? These are all microtubules. What the microtubules are going to do is they are going to separate the two copies of chromosomes. දෙකට කඩනවා. So දෙකට කඩනවා නෙවෙයි, right? They are going to just separate the two copies, right? So that one copy can be given to each of the daughters, right? So that's going to happen next. You can see. So that is going to make, I mean, the cell is going to get elongated because the mother has to divide into two. Then, the DNA, the two copies have been separated. Then they are going to the opposite ends, and at the same time, can you see? There's two nucleuses, we call them nucleoli. Two nucleoli are being produced, the nuclear envelope is being reformed, and the cytoplasm got separated. Hold on. Right, where were we? Oh, nothing. Right, so the nuclear envelope got reformed and the cells separated. And you got two complete daughter cells. Okay, that is a summary of mitosis. If you watch it with the proper sequence, okay, the chromosome copies are going to be separated and they go into the two opposite ends, and around them two nuclei are going to be produced and you're going to have two complete cells. Simple. Meiosis, if I just quickly tell you what's going to happen in meiosis, meiosis has two parts, as meiosis 1 and meiosis 2, almost exactly the same. At the end of meiosis 1, you are going to have two cells. Okay? At the end of meiosis 1, you are going to have two daughter cells. At the end of meiosis 2, these two cells are going to divide again to form four cells. That is meiosis, the entire process of meiosis. So at the end of meiosis, you are going to end up with four daughters. At the end of mitosis, you are going to end up with two daughters. Right. That's it for now. We will see what I can explain with the software. I explained to you what my wallpaper is, right? Yeah? Did I explain? Yeah, this is a 3D CT scan. Okay, of a, I mean, right, he's one of my patients, so he had a motorbike accident, right? So you can see the damage caused by the accident. He was not wearing a full face helmet, so he collided with the back of a lorry, and you can see these maxillary fractures are there, and then my jaw එක මැදින් separate වෙලා තියෙනවා, right? There was a what we call as a mental කියන්නේ එතන, that's called the mental protuberance. It has been separated there. Okay. So that's what happened there, right? So it's a sort of a very interesting thing you can do with CT scans. You can generate a 3D image. Anyway, let's come back to okay, take the resource book. Online people, can you hear me? Yeah? Okay, right. Hi. So before I actually get into the proper cell division explanation, I want to give you some background knowledge. I want to give you some background knowledge. Without this core knowledge, you will not be able to understand. So listen carefully. We are produced by the combination of how many parents? How many parental organisms do we need as humans to become fertilized or to be conceived, to be created? How many parents? Two parents. Yes. So you have a mother and a father. Ne? So you, if you look at your genetic composition, you are exactly 50-50 sort of a combination of your mother and father. Exact 50-50. Right? So then why are you more සමහරවල්ලාට කියනවනේ අම්මා වගේ වැඩිපුර තාත්තා වගේ වැඩිපුර. Then if you are exactly 50-50, how do you look more like your mother or like your father? That is something that happens genetically. That means like with the knowledge of genetics you can explain. But if you take one of your cells and analyze, you are exactly 50% your mother, exactly 50% your father. Right? So that knowledge we need to know. So if I, right, so this core knowledge I have to give you before we do anything. Okay. So when you were created, there were two cells involved, right? There were two cells involved. There was an egg, right? There was an egg that was given by the mother, usually big cell. Okay, this is the egg cell. Clear? Then there was a sperm. Usually very small, but I'm going to draw it like this, right? This is a sperm. Got it? These two got together by the process of fertilization and produced a single cell that is called as a zygote. Okay, right? The zygote, a human zygote, has how many chromosomes? Chromosomes kiyak තියෙනවද? 46. 46 chromosomes. So you can do a sort of a genetic analysis technology. You can do something called a karyotype. Okay, that means you can take one of your cells, you can take all the chromosomes in there, you can arrange it from the chromosome number 1 to chromosome number 46. I think in the very first tute, in the first page I gave you a similar diagram. The interesting thing you can do is you can, when you are doing that karyotype study, you can arrange them in a way so you can pair up the chromosomes you got from the father with the mother. මෙහෙමයි වෙන්නේ එක. So for example, let's say your chromosome number 1 looks like this. Huh? Chromosome number 1 out of 46. Okay, this is, let's say, a chromosome you receive from your mother. Right? There's another chromosome that looks, I mean, it's similar in shape and size and length and everything to this one you got from the father. Right? So we call this our first chromosome pair. First pair. Right? Then let's say there's another chromosome that is sort of ටිකක් මීට වඩා කෙට්ටුයි දීගයි මෙහෙම. All chromosomes have different shapes. Right? Then scientists found out there's another chromosome that you got from your father that is exactly the same shape, size and length. මේකට අපි කියනවා chromosome number 2 කියලා. Then let's take one more example. තව එකක් ඉන්නවා ටිකක් කොට මහත එකක්. Chromosomes. Right? The chromosome අපි හිතුවට they don't look all the same, right? This is like the classical diagram. There's another one you got from the father that is exactly kind of like that. Right? So for example, imagine the make it a bit of a complicated genetic theory. Right? Imagine in this chromosome here මෙන්න මෙතන you have the gene කියන්නේ මොකක්ද? A piece of DNA that is going to determine something. Here this box contains the set of genes that is going to determine maybe your hair, maybe if it's curly or straight. Right? So your mother has curly hair, your father has straight hair. May they interact, you are going to get either curly hair or straight hair. That we don't know. So forget about that. What I want to tell you is, you got from your mother 23 chromosomes. Okay? You got from your father another 23. Okay? So in this egg, you had 23 chromosomes. Online people, listen carefully. Are there? You had 23 chromosomes. In your father's sperm, so sperm එකේ මෙම head එකක් කියලා තියෙනවා. Head in there, there's a nucleus. You had, you got from your father 23 chromosomes. Okay. So you can draw this until you get your 22nd pair. 22nd set එකනකන් you can draw this. 22nd. 22nd එකට යමක් කර චූටි එකක් right 22nd can be something chooty like this. We don't know. Right? Exact karyotype again you can understand. Why 23? Why not 20? Sorry, why 22? Why not 23? Because the 23rd pair is special. Don't know, right? If you are a girl, from your mother also and father also, you receive the same, right? If you are a girl, from your mother you received an X chromosome, and from your father also, මම එක blue වලින් අඳින්නේ because it's from the father. Okay, you received an X. This is, if you are a girl, this is your 23rd pair. You know දන්නවනේ the sex determination, right? If you are a boy, from your mother you got the same X. Okay, but from your father you got a චූටි එකක්. That means a chromosome. That is the Y chromosome. Okay, if this is a boy, this is the combination. If it's a girl, these are both 23rd chromosomes. This is female. මේ O level උගන්වන්නේ හරිද? Right? Male. Okay, so we call these pairs homologous pairs. ඒක දැනට අමතක කරන්න. Right? So what I want to teach you right now is, we give a name to the condition of having such two sets. මෙම sets දෙක දෙක ගානේ තියෙන එකට we give a name. That is called being diploid. Having two sets. You give a symbol for that. That is called 2n. Done, right? 2n. Right? Having only one set is called haploid or half. You indicate that by the symbol n. So your mother's egg was n, your father's sperm was n, haploid. So two haploids got together to form a diploid. Clear? Then now you know, after they made this sort of a zygote, this is you. You originated as a single cell. So how did you become the billions and billions of cells that you are right now? By mitosis. This single cell divided by mitosis again and again and again and again until it became billions and billions of cells. So what's going to happen is, this zygote is going to divide by mitosis and produce two daughters. The two daughters are going to again divide each by mitosis, going to get four daughters. මේ 4 8ක් වෙනවා 8 16 32 කොහොමම exponentially the cell number is going to increase. That is mitosis, how it works. So how did you got these two haploid cells? That is by meiosis. Okay, so always remember, meiosis only happens when you are making haploid cells. What are your haploid cells? Gametes. Exosperm හදද්දි විතරයි you do meiosis. Any other time where there is cell division happening in your eukaryotic body, that is mitosis. Okay, muscle, I mean, your sort of, you have a wound and that there's tissue regrowth, that is mitosis. Yes, in your digestive system so many damage is happening that cells are being regenerated, that is mitosis. You are getting bigger, growing taller, that is all mitosis. Mitotic cell divisions are happening everywhere. Meiosis is happening only when you are making either exosperms that are gametes. There or not? This basic idea should be there. Okay, got it? Clear? Okay. Then we are going to move on to the proper cell cycle, that is with the study of mitosis. Any questions? Okay. Maybe preliminary sort of basic information should sort of, I mean, you should remind yourself of that. Clear? Okay. Ne? Right. Okay, come to the resource. Cell division. Okay. Page number 39 onwards. Okay. Let's read. The cell cycle and the process of cell division. There's a definition. Okay, we'll start with the sequence of events that take place in the life of a cell from end of one cell division to the end of the next cell division is referred to as the cell cycle. Okay, so so we think the cell cycle means how a cell divides. No, your entire life is not your reproduction. Cell division is a cell is reproducing. But a cell has another life. Yes, a cell lives a nice long life, and at one point it is just going to divide. Then that divided cell is going to have its own life. Then at some point it's going to divide. It's kind of like your life, right? You are going to be born, and then you are going to grow up and going to become adults and give rise to another cell, then going to live your life, right? That's what happens. Okay, so the cell cycle is actually the life cycle of a cell. Okay, a small part of that has a cell division, right? That's what this definition says. Highlight can sequence of events that take place in the life of a cell from end of one cell division to the end of the next cell division. Okay, cell cycle. After cell cycle, like, because it's a cyclic process that happens again and again and again, you can indicate it as a one, I mean a continuous loop. Okay, so okay, what is the definition? From the end of one cell division to the end of the next cell division, no? Right? End of one cell division to the end of another cell division. That is where, that is what we call as the cell cycle. So imagine the same diagram is given in your resource as well. This is where a new cell was produced. Okay? This is the birth of a new cell. Right? This is where that cell divides. මෙහෙම full round ඇවිල්ලා this is the point where that cell was born. It's going to live its life and then it's going to divide. Okay, this is the cell cycle. Actually, the cell division is only a very small part of its big life. And but we are going to learn about this whole cycle today. Got it? Right. So it's given. This is where the cell divides. That is called as cytokinesis. Okay, cytokinesis. that is part of the cell division. Okay. Then there's a very important part that is called the mitotic phase. That is where the nucleus doubles so that it can be given to two daughters. Right? M phase. We'll talk about that. What I want you to pay attention is the rest. You can divide the rest into three pieces. Okay? You can divide the rest into three pieces. So today I'm only going to talk about, I'm not going to talk about cell division actually. I'm going to talk about what the cell does when it's normally living. සාමාන්‍යයෙන් cell එකක් විදිහට සමාජයේ ඉන්නකොට මොනවද කරන්නේ? Okay? Right. It has, when it's normally living, when it's not dividing, it has three sort of phases. They are called G1, S, and G2. Okay? Got it? This is what we are going to talk about. Collectively, this G1, S, and G2, මේ තුනට we give it a name called the interface. Interface කියලා කියනවා. That means all the things that is happening to the cell when it is not dividing, living its life. සාමාන්‍ය විදිහට ඉන්න. Got it? Understood? Okay. Okay, come to the resource again. Let's keep reading. Next page. Eukaryotic cell cycle. Eukaryotic cell cycle may divide into two major phases as the interphase and the mitotic phase or the M phase. Okay. So we'll call this as mitosis. Altogether we call this M phase. Okay, interphase and M phase. So your eukaryotic cell cycle has two phases. As the big interphase, which is actually about 90%. 90% of the cell cycle, then it's sort of bigger than that, right? 90% of the cell cycle is actually interphase. And the M phase, that's where the actual cell divides, is 10% of the cell cycle. That means, I mean, in the lifespan, imagine the cell lives for about 10 hours, right? Without dividing, 10 hours now. 9 hours is spent in the interface. Only 1 hour is spent dividing. Imagine as a human, he lives 100 years, 90 years he lives normally, and 10 years reproducing. Okay? Dividing into two. Got it? So interface is the longer phase. Right. Read, okay? Interface is the longer phase. It covers about 90%. Interface could be divided into three phases. Interface could be divided into three phases. They are called G1, S, and G2. G1, S, and G2. Let's read, okay? Let's read and we'll take down small notes in the diagram tute as we are after discussing, okay? G1. In this phase, synthesis of proteins and production of cellular organelles leading to cell growth occur. Proteins essential for the S phase are produced. Right? So G1 stands for the first growth phase, or you can say first gap phase. Okay? So then imagine after the cell division is over, chuti cell just produced. So that cell is going to grow, get bigger, right? It's like you, when you were just born, it's just going to grow. So G1 is a growth phase, right? So you can say what happens in G1. Okay, if I G1, what's going to happen? Protein synthesis. What else they say? Protein synthesis, production of cellular organelles. What else? Then proteins essential for the next phase. Yeah, ඊළඟ phase එකට last වෙනවා, right? Is going to prepare for the next phase, which is the S phase. S is short for synthetic phase. Okay, synthetic phase. Very important phase. He is going to prepare for the future by what? In the future, what's going to happen? It's going to divide into two. So it has to copy its DNA so that it can give it to the two daughters, right? Then at the end, so that in S phase, what's going to happen? Very, very important, DNA replication. Then if you have two siblings, maybe your father builds two houses, no, then no need, right? The cell prepares for the upcoming cell division by copying its DNA replication. So that is not enough, no? You have to wrap that DNA around protein so that it can be packed. So histone synthesis. Histone protein, then the unknown histone, histone synthesis occurs. Then DNA produced, histone produced. DNA wraps around the histone, very, very important, to produce chromatin. Form chromatin. That is what happens in S. Okay. Then you come to the final phase, that is G2. Final phase of interphase. Right? What's going to happen in G2? Further, I mean, it's very similar to G1 actually. Right? G2. Okay. Protein synthesis. Organelle production. What else? Very, very important thing happens because the now we are very close to be divided. Divided in the what's going to happen is, what do you need? You need the chromosome separating machine. What is that? The two centrosomes. Okay. So very important thing happens, that is centrosomes duplicate. They become, you produce two centrosomes. So you can say centrosomes duplicate. Centrosomes duplicate. Very important, let's say, incidence that is going to happen in G2. Okay, right. Let's take this down in the diagram tute. Okay, take out the diagram tute. Page number 7, I have given you some space. Okay. Cell cycle කියන එක divided, you have the topic itself, no? ඒක දෙකට බෙදන්න as interphase and M phase. Interphase එක, get it into G1, S and G2. Write down the important points, but please save space. තව ලියන්න දේවල් තියෙනවා. Okay? Because according to me, this is like a pattai duck, right? Okay, right? Right? So you have to write a lot of things in this space. So manage your space. Write down the important things that is happening in G1, S and G2. G1, protein synthesis, production of cellular organelles. Make a full video, okay? Production of cellular organelles. මේ දෙකෙන්ම වෙන්නේ මොකද්ද? Right? Cell growth. Cell growth. Then S phase, DNA replication, histone synthesis and formation of chromatin. G2, protein synthesis, cellular organelles, duplication of centrosomes. Very, very important step. Okay. So there's a question. What if there was an abnormal incident forming chromosomes? Yeah, mostly Abnormal incidents are prevented by there's an internal mechanism in your built into your cells that will talk. That is the immediate topic that we are going to talk about. Okay. Okay, done. So while you're writing, as I always say, think about what you're writing. Okay, Dhamma Padanka. Okay, try to memorize it right now. What's going to happen in G1, S, and G2? Haridu. Then we come to that question: what's going to happen if something goes wrong? Because you can see, particularly in this S phase, something very, very critical happens. What is that? The DNA is copied. You have, I mean, you can say you have 20 to 22,000 genes. A single gene has thousands of nucleotide bases, nucleotides, a single gene. You have 20,000 of that. So imagine the length of DNA you have. Several sort of these sort of what we say as explanations are there. You can wrap your DNA twice around the earth if you sort of lay it as a straight strand, right? That's how long that is. Right? But that DNA has to be copied. That much DNA has to be copied. So when that DNA is being copied, an error can occur. Very common, right? Imagine you are making 20,000 photocopies, few errors are there, yeah. But your body doesn't tolerate that because it's too risky. For example, a small problem in one of the cells that are produced by the thousands of cell divisions that is happening even right now in your body can be the start of what? A cancer. Yes. A cancer starts due to one abnormal cell, a single abnormal cell. If a single abnormal cell is produced, that abnormal cell will start to behave abnormally. It will absorb all the nutrients around it. then it will start dividing rapidly, then it will invade other cells, it will spread everywhere in your body, and eventually it can kill you. That is like the worst case scenario. There are abnormal cells that just form a ball of cells, right? Uterine fibroids in the uterus of females, you can get these balls of abnormal cells, fibroids. Yes, those kind of cells are abnormal balls of cells that are not actually cancers. We call them as tumors. Whatever, mitosis has to be very properly controlled, otherwise it could be dangerous. Then how does your body regulate mitosis? How does it keep an eye on? There's a special, I told you, quality control system for the entire mitosis process. That is done by what we call as checkpoints. Checkpoints. That means whenever a cell is going through all these G1, S and G2, there are, in those phases, that cell is evaluated. Put the interview kalla balana meya honda ide. Has he completed everything that has to be done in G1? Has he completed everything that has to be completed in S? Properly complete kalla tiyena will be assessed. Ogolna shishyath liyena O level liyena right? That's how you test yourself. ඒ වගේ there are checkpoints. Read, come to the resource book. There are cell cycle controlling checkpoints available at G1, G2 and M. Okay. To ensure that the cell is ready to move for moving into upcoming phases of cell division. So in your diagram tube, I have given that cycle. Can you see? Page number 7. You can see the checkpoints. G1 checkpoint is there. There's a G2 checkpoint. Then there's a M checkpoint. Right. So at the end of G1, sort of a flag, right? This is one checkpoint. At the end of G2, there's another checkpoint. And before it divides immediately, there's a checkpoint here. Those are the 3 checkpoints. In those checkpoints, the cell is assessed. Check if everything is ready to be divided. If everything is not ready, it won't divide. Got it? Metin? Some cells receive a go-ahead signal at G1 and it will usually complete G1, S and G2 and then divide. G1 ekedi it has to receive a go-ahead. Green light ekak enna one, otherwise it will not be allowed to continue. Got it? If it does not receive a go-ahead signal at that point, it may exit the cycle, entering into a non-dividing stage called G0. Very interesting. Okay. If the cell fails, okay, for example, O level fail huna ke liye, what are the options that you have if you failed O level? You can rewrite the exam, right? So this G0 is a very interesting phase. So imagine if you failed here, the cell was rejected and it will be entering into something called G0, non-dividing. Okay, G0 non-dividing phase. A cell in this G0 phase actually has 2 options. Okay, it can correct itself. It can correct itself here. That means you can study for the exam and face the exam again. So until that, you are put into a G0 phase. Okay, that is one thing. The other thing is some cells are permanently in G0. There's a transient G0 and there's a permanent G0. Again, somehow then O level fail job they don't want to face the O levels again. They just go into jobs again. Sorry, directly into a job. Okay. But some people, they want to pass O levels, so they study and face the exam again. So 2 things can happen in G0. One, after they have completed the things that has to be happen at G1, they can continue the cell cycle. They are allowed to go ahead. Or they can remain in G0 forever. Kavadawat I divide wenna. Yes. They are never going to enter the cell cycle again. Later they will somehow die off without ever dividing. In your resource, they have given some examples for cells in G0. Monadh dila thi? Most of the cells in the body are actually in G node, which is kind of not accurate. But the examples given are what? Nerve cells and muscle cells. Your nerves and cells and muscle cells are actually, they are cells in permanent G node. They are never going to divide. Nerve cells, muscle. Okay, they never divide. Once when you were a baby, when your brain is being formed, there was some cell divisions, and then your brain, all the neurons were produced. After they divided and produced, became the neurons that you have right now, they are never going to divide again. If you have a stroke right now and part of your brain dies, are you going to regenerate that? No, never. If you are a chronic alcoholic that drinks heavily for a long time, your nerves are going to be damaged. Are they going to be regenerated? No. Okay. If due to a certain accident, part of your thigh muscle has to be removed, is it going to grow back? No. Muscles are never going to divide again, skeletal muscles. If you have a heart attack and part of your heart muscle dies, is it going to be regenerated? No. Muscles don't divide. They can only get bigger. Muscle cells don't divide. Okay? They only get bigger. When you go to the gym, what's going to happen? Muscle cells are only going to get bigger because of the sort of the increased protein production. Okay? They are not going to divide. They are permanent cells. They are in permanent G node. Got it? But G node it's not always permanent. I told you, they can be temporary. So they can re-enter the cell cycle once they are qualified. Understood? Okay. The මෙතන නැති තව එකක් තියෙනවා. For example, if a cell never able to rejoin the cell cycle because it is damaged beyond repair. මොකද කියන්නේ? If it's damaged beyond repair, we talked about this when we are talking about lysosomes. If a cell is damaged beyond repair, මොකද වෙන්නේ? If a cell is damaged beyond repair, ගොඩ ගන්නම බෑ. වාහනයක් like completely crash වෙලා, you can't repair it. What's going to happen? You're going to destroy it. No? In your body, there's a destroying mechanism that is called what? Autolysis. Autolysis trigger. Cell is going to die. There's nothing going to happen. Such things are happening in your body right now. Cells are being killed. Cells are being put into G node. Cells are being converted to permanent right now. Okay, that's the sort of the beauty of this G node. That's how you are not getting cancers at any given moment because of these cell cycle regulators. There are specialized proteins to do this. Okay, cell cycle එක monitor කරන්න there are special proteins. අපි ඒවට කියනවා policeman of the cell scale. Right, he comes and checks everything. Right, you will learn about those not in A levels, but understood, clear. Okay, then page number 7 එතන අපි ලියාගමු in the diagram tute. Write down a point. Write down a point. There are cell cycle controlling checkpoints. There are cell cycle controlling checkpoints. There are cell cycle controlling checkpoints. There are cell cycle controlling checkpoints at G1, G2, and M phases. At G1, G2, and M phases. G1, G2, and M phases. Next point. If the cell doesn't receive a go-ahead signal, if the cell doesn't receive a go-ahead signal at G1, if the cell doesn't receive a go ahead signal at G1, it enters into G naught. Then G naught කියන එක put a small subtopic as G naught, write under it non dividing, then divide it into two as transient and permanent. Then under permanent, write down the two examples, nerve cells, muscle cells. Write down the two examples, nerve cells and muscle cells. Nerve cells and muscle cells. Nerve cells and muscle cells. Okay. Okay. Any questions? Okay. Right. So these days we are doing a forensic appointment. What do you mean by forensic? It's called Adhikarana Vaidya. That is a sort of the branch of medicine related to sort of the legal matters, right? So legal matters can be anything from a simple husband and wife fight or a school sort of between two school children or to a complete rape case and a murder or a serial murder, whatever, right? Mysterious circumstances of death, right? Death or injury, whatever, right? So it's an interesting field. So I just came today from the mortuary, right? So in our hospital mortuary, we have to observe autopsies. Autopsy means what? When a person dies under suspicious circumstances, uh, they can request, or there's in Sri Lanka there's a person called මොකද ISD කියලා අහලා තියෙනවා, inquiry into sudden death. හදිසි මරණ පරීක්ෂක. අහලා තියෙනවද? Right? He can order, okay, some person has died in a home and this death is suspicious, poison කළාද දන්නේ. So they can order the doctors to cut the body and check. Or you find a dead body somewhere outside, කානුවක වගේ කොහේ හරි තිබිලා, then you don't know how he died, if it's a murder or not. Then we have to cut open and see. There's like very specific techniques to do that. You open the brain and take the skull, open the skull and take the brain out, and the lungs and the heart and the intestines and everything. So recently we had a very interesting case in forensic. There was very old means old dead body. That was a person that disappeared during Ditwa, the sort of the entire heavy rain season. So the body was found. Okay, so I want your help. So I'm going to give you the details of the body. You can tell me how he died, right? Circumstances. So, okay. So we were shown some photos, right? We were shown some photos of the dead body on the autopsy table. It was an old case. On the autopsy table means in the mortuary you have these metal tables with proper drainage so that blood can drain. So there was a person on the autopsy table, right? Lying on the autopsy table like this. Okay. So he was wearing a t-shirt, only a t-shirt, nothing at the bottom. Naked from only a t-shirt. He was wearing only a t-shirt, right? And when we look at his face from the nose, there was some froth coming out. Bubbles, bubbles, from the dead body. Right? That is a clear sign that the person died from drowning. Right? If a person died from drowning, right? වතුර ගිල්ල මැරුණොත්, even after death, because his lungs were flooded with water, that water is going to come out mixed with blood and sort of mucus and everything. දිගටම එනවා නහය. Right? When you wipe it away, some more is going to come out. That is a clear sign of the person died by drowning. Okay, so the person died by drowning. That is very clear. And he's covered with sand. හැමතැනම වැලි වලින් කවලා තියෙන face and the clothes and everywhere. Okay? Then we were asked, okay, so this dead body, our professor didn't tell us where the body was found. Okay, so we told, okay, this person died by due to drowning. That is clear. Then he asked, okay, why wasn't he wearing anything under when the body was found? Body was found like this. Why wasn't he wearing anything to the lower part? Okay, so we said, okay, definitely naanna gihilla newe, right? People don't, men particularly, they don't bathe with only wearing a t-shirt, Okay, so probably not bathing. That's what maybe malu allana gilla, fishing. Okay, or was traveling on the road and was caught into the flood, Gangwatura. Then, but still, why not nothing underneath? Then I won't say who said, not me. Maybe he was wearing a sarong. What do you think? Yeah? Very easily dragged away, Sarama Galaveno and Niga. Got it? Then the professor asked, okay, but why wasn't he wearing any underwear? Right? So, again, it's not me at all, because that's how you wear a sarong. Okay? So, one of two things could have happened there. Either I would have been thrown out or the professor would have laughed. He's a very serious one, so he rarely laughs. Okay, he laughed. Okay, දෙකෙන් එකක් වෙනවා එළියට දානවා කියලා හිතුවේ, but no. Yes, so he was wearing a sarong. So what can you say about the circumstances? Was he going somewhere important? No, he was probably close to his home, ne? Probably close to his home. Or what? What are the other circumstances that he was only wearing a sarong? Think about men, right? Probably drunk. Eh? Probably drunk. Yes. So now you know sort of the scenario of how he died. Okay. Then we were taken to the mortuary. Okay, we were told to examine. Then balanna kiyan kiwa. So on the table it was there. So what we did was, so we examined everywhere. Nothing. No damages. Nothing in the body. We found out that his hands and his sort of the feet are covered in mud. Mehema atwalai, and on the feet there is mud, but not anywhere in the body. There's just sand in the body. Okay, there's mud in the hands and feet. Then we turned the body other way around. Anit patta hara. Okay, then we found out he has scratches on the back, particularly around the shoulders, on the back. He has scratches. So then the question was, now you have to, based on these, what you can find, where do you think the body was found? What do you think? The person died by drowning, and he has scratches on his back, and he was probably drunk, and he has mud on his feet and hands. More again. How do you think he died? How do you think he died? More again. Yes? Any online people? There's a subject related question. What happens if the cell doesn't receive a go ahead G2? Okay, I'll answer that. Okay. What do you think? Any idea? What again? Where did he drown? Go ahead. Yes. Actually, he drowned in a mehema on the road. If this is the road, there's a mehema bokkuwat, only about 1.5 foot tall. මෙහෙම බෝක්කුවක්. So the body was found inside that. The dead body was found inside this bokwa, and here up everywhere it's mud. Everywhere is mud. The body was found inside this. So what has happened? This person was walking home drunk after going to the bar, heavy rain, and the entire road was flooded, so he couldn't differentiate between the outside මේ පැත්තේ කුඹුර and the par. Right? Now කුඹුරට stepped in, and there was a massive whirlpool of water, high sort of because of the rain, and that water was going through this sort of this drain, right? බොක්කෝයට massive. There's a massive current of water. So he was dragged feet first. Feet first, he was dragged into the bok. So he was facing down like this, and feet first he was dragged into because of the massive water speed. Got it? So because he was drunk, he was not able to properly control everything, right? So he used his hands and feet to try to climb out, but because it's a very short roof, it okkama hiru, right? So the body was found inside there. Okay, and it took the family about, I think, the person was missing for about a month or something until the body was discovered and was brought to the mortuary, and police was notified and everything happened. So the body was in the fridge for a long time. Okay, so that's one of the things we encounter every day in forensic, right? Sort of Sherlock Holmes stuff sometimes, but sometimes it can be depressing. You don't get to meet people. It's all about sort of putrefied dead bodies, right? Anyway, so if you are interested, maybe later you can think about once you are in medicine. Okay, but the pay is not very good because you can't do a private practice. There's no private mortuaries, okay, right, to do this sort of investigations. Okay, you have to work in the government. Anyway, we'll do one MCQ and be done. There's a question: what happens if the cell doesn't receive a go ahead at G2 checkpoint? Usually this G2 is a transient checkpoint. G2 is a transient checkpoint. It is not that much of a serious checkpoint because at G1, that is where you can put cells into either autolysis or permanent. At G2, you will keep the cell until it is properly ready and it will receive a go ahead signal sometime later. G2 is not a very serious one. Okay. Right. Abhi ek MCQ ek karumo. Okay. Come to the question tube. Come to the question tube, page number 5. Sorry, page number 5. Question number 4. Taggalagan. Before the rain, you can go home. Okay. Question number 4. Ne? Yeah. Page number 5. Question number 4. Taggalagan. Everything is given in the board actually. Okay. If there are checkpoints, then how are cancer cells formed? Because of the sheer number, right? You have thousands of cell divisions happening right as we speak. So there's a chance one cell escapes. Yes, there is a problem with having billions and billions of cells. One can become can go undetected. That's how even there's a question: did you encounter serial killer? That's how serial killers are formed, right? When the population is massive, one person can hide in that massive population and keep doing illegal things. So a cancer cell is like that. When there are trillions of cells, one cell can become abnormal. So the checkpoint is preventing out of the trillions of cells, it's preventing thousands of cells becoming abnormal. Ekak dekak can escape. Got it? Okay, taggalakan, taggalakan. In Sri Lanka, there are not much serial killers. Okay, not much. Usually, they are serial killers are sort of confined to sort of rural areas, and we don't usually get that sort of cases. In sort of our urban areas, serial killers are usually pathal assassins. So they are considered as serial killers, right? They are paid killers. So that's something different, but still serial murders, but with the money as the motivation. Nothing sort of not like in other countries, right? Cannibals like Lanka. Cannibals means what? People who kill other people to eat them. That's very rare in Sri Lanka. There are some sort of unsolved cases. Kotaketana Kila, there was a series of murders that happened over 8 years. I think 15 women were murdered, but there was not a permanent sort of culprit was not never found because we think it was several people, right, who are sort of are imitating the serial killer, right? So that is very common in Sri Lanka, right? Anyway, right. Question number 4. DNA replication G naught. Does it happen in G naught? No. G naught is a non-dividing phase. Yes. In front you can write G naught is non-dividing phase. Second one, synthesis of proteins. That happens in what phase? S. I'm sorry, G1. I'm sorry. G1, that is correct. No? Synthesis of proteins, that is a correct answer. 3, chromatin formation G2. No. You can correct it. Chromatin formation happens in S phase. 4, production of cellular organelles S phase. It can be G1 or G2. Ne? You can correct it. Can be either G1 or G2. Duplication of centrosome G2. Ne? G2. Correct it. G2. Right. So we'll finish mitosis next week. Okay. So do the MCQ homeworks. Okay. So we'll discuss those next week. And now you have a studyable quantity. Then padankan pulang quantity kak you have. Entire cellular structure you can study, chemical basis you can study, right? So allocate at least maybe 15 to 20 minutes. Okay. Hopefully you are studying maybe 1 hour per day now, 1 hour just to take out the books, the 3 subjects, physics, chemistry, bio, tuna poddak eliyata aragen 20 minutes per subject. Chutta padanga, chutta. Minimally, okay? Just read what you learned last week. And online people, small announce online for you also. If you feel like you need to go through everything from the beginning, Haritha, I'm going to start a special series that is going to happen purely online, probably on Monday, bit late at night. That is going to sort of bring you up to speed to write at this point, starting from the very beginning, from the introduction to biology. Within 4 weeks, 4 sessions, I can rapidly bring you up to the level that we are doing right now. Okay, so that's going to be a free session. If you missed the earlier lessons or you are just thinking of switching to bio, you can join that. I'll send all the details to the group. Okay. Right. Yeah. That's all. Okay. Then good night. You can go. Hello, people. Any questions? Good night. Question about the medicine. Okay. Yeah. To be honest, our final results. You have to do a final exam there and graduate. You have to come back here and do a exam here as well. So if you just graduate from Sri Lankan faculty, you don't have to. You are going out with the MBBS from the final exam. So the reality is that the foreign graduates that are coming to Sri Lanka, they struggle with the exam, which is much higher standard. The year two. So they have to specialist. You mean a consultant? To become a consultant, that's a post-graduate. So the post-graduate is completely different. So you have to either, if you, I mean, to complete your internship here, you can apply, you can do the, just do a part to any specialty. Consultant direction is two parts. There's a part one and a part two. So it takes at least about 5 years to complete. So you can complete those two here, but definitely you have to go abroad for the foreign. Definitely you have to. Yeah, there is. No, you do the exams here, then you go there for the training. Then you come back, and as a fresh consultant, you will be put to somewhere different far away. You have to work in a distant hospital for maybe 2 to 3 years before you are allowed to come to Colombo or somewhere you go. So the average age for consultant is about 35. So if I do the MBBS in Malaysia, doing some work for specialties and products, so can I actually do the exams? You have to do the ERP even if you are consultant. Even if you are consultant. So that is going to take, so after your A levels, you are going to go there. So that is going to take at least 8 years to become a consultant and all. So if you come back here, you have to do that exam because you have to need the license here. To get the license here, you have to do the exam. Another question, since you mentioned forensics, I have a relative of mine who is have you heard the Chirath scientist? Doctor, his wife, Asen. Asen is it's a very old thing, okay? Very old. Yeah, yeah. So, the case said that they found he was a professor of Asen in his clinic. So, why would the doctor be professor of Asen? It's because with our license, we can purchase Asen. That's it. It's not exactly prescribed Asen. I don't know any modern medication for Asen right now because with our license, we can purchase. No, I don't know. I don't think if it's in modern medicine, there's a place for Asen. It's another case. Yeah. If you have any files, you can send. Yeah, absolutely. So why did you catch the Asen poison very low? It's very rare case. Asen poison is very rare. Because we sort of our main poisons are sort of usually are pesticides. So they are easily found in the stomach sort of contents. But arsenic usually travels to the hair. They are in the liver particles, nails. So we don't check routinely. That's why it's easy to miss it. In other countries, arsenic poison is common. So you got arsenic is very common. Okay. Okay. Okay, online people, then good night.